Good afternoon. 200 participants on Zoom, about 80 participants on site. Heartfelt welcome to this program symposium brought to you by Mistra SafeChem. It's such a pleasure to have you with us. So my name is Ulrika Edlund. Together with Professor Lennart Bergström, we will be happy to moderate today's session and walk you through this really exciting afternoon, packed with interesting talks and a lot of information. Before we get started, there are a few notes that I please ask you to listen carefully to. There will be many presentations, and I'm sure that it will spur many questions. We will take questions after each presentation if we have time. For you who are here on site, we also, of course, encourage you to talk and discuss and ask a lot of questions during the intermission. If you are participating on Zoom, please type your question in the chat. We will monitor the chat and we'll be happy to moderate your question for you. If you're participating on site and wish to ask a question, please just raise your hand. We will bring a microphone for you. So don't speak until you get the microphone, because otherwise people that are participating on Zoom will not be able to hear your question. Few other notes. Please respect that smoking is not allowed anywhere on site. Please also know that there are 4 emergency exits in the room. There are 2 in the back and 2 on each side in case we for any reason need to evacuate. Please calmly leave the building and use the emergency exits. We also ask that you please do not take photographs. We have an in-house photographer. She's of course allowed, but otherwise, please know that this session will be recorded and you can watch all the presentations on video afterwards. The recording will be posted on the Mistra SafeChem website for your kind pursuit whenever you wish to go back. And that is, of course, also for you who are participating on Zoom. So no need to take photographs right now. Should you need a restroom, please find them if you use the back exits and then make a right and then a right again around the corner. You will find restrooms, preferably done during the intermissions, of course. Would you like to connect to Wi-Fi? Please know that there's a free Wi-Fi in the house. It's called AZ Guest. You do not need a password to access it. You can just log on if you wish. And with that said, I think that we can move on to the exciting part of this afternoon, right? So this program is brought to you by Mistra SafeChem. And a few words about Mistra SafeChem. This is a research program, name implies, it's funded by Mistra. It has a large consortium of partners that cuts across industry, academia, and industry. It started the first phase in 2020 and is therefore halfway through the first stage. After 4 years, Mistra will give you the opportunity to apply for a 4-year extension. If so, that will be 2024 that the next stage can begin, if so. What we do, or the vision that we share, all of us, is to enable and promote the expansion of safe, sustainable, and green chemical industry in Sweden. That's what connects everyone participating in or interested in Mistra SafeChem. And that is what we will explore today at today's agenda. Should you like to know more about Mistra SafeChem, please go to the website www.mistrasafechem.se and you will find a lot more. You can also find an entry to Mistra SafeChem information on the homepage if you go via IVL, Environmental Institute of Sweden, who acts as the host for Mistra SafeChem. All right, so we have a fascinating program for this afternoon, packed with many prominent speakers. We are here on site at AstraZeneca. Of course, we are very grateful for AstraZeneca for hosting us, allowing us to come here, and for also telling us more about all the exciting work that you do when it comes to transitioning the process to sustainable chemistry. And we have an expert on the topic here to tell you a little bit more about AstraZeneca's activities in this field. So I welcome to the stage Malin Lemurell. There you are, Director and Head of Medicinal Chemistry. Malin will tell us a little bit more about the activities that you do. Please.
Thank you very much. And the true expert in the room is actually Stewart Owen, who will speak later on this afternoon. But we are very committed here to drive the sustainability and green chemistry across our chemistry community within the company. So you should feel most welcome. And if I go back, you're here now today at the site of AstraZeneca in Gothenburg, which you see on the picture. And this is one of the 3 global R&D sites we have. And we have, in addition, another 8 R&D centers and offices around the globe, and 28 manufacturing sites. So it's a huge community, and we really try to drive the green chemistry and sustainability jointly. At the site here, we are very proud that we have the full value chain from very early discovery all the way up to running clinical trials and doing toxicology studies, and also manufacturing and production of the clinical trial tablets. The full production for our marketed drugs we have at the other sites, which is one of them are in Stockholm, in Södertälje. We have more than 2,800 employees at site, and we have representation from more than 70 different nationalities. And we have a very high education level, which makes it extremely interesting and fun place to work, with a lot of creativity and inspiring colleagues to discuss with. We're also very proud to also let you know here, in the context of today's topic, that we have now removed more or less all carbon dioxide footprint from emission here. So we only rely on renewable energy sources, etc., for driving this site. And also we are transitioning the full car fleet over to electric cars, etc. And that's just one step on our sustainability journey. So at the site here, you can see in the orange area, this is the current site, but we're also investing a bit south together with the GoCo Health Innovation Center, which are buildings. So if you come along the highway, you will see that we have new buildings coming up there, and there will be a big innovation and life science cluster here, which will double the size of research in life science in this area. Also, today we are hosting HealthWorks and Oligonova in these buildings. And in the center of our site today, we have more than 28 biotech companies who's actually driving their research, and they are located in the center of our site, so we can have good discussion and interactions with them. And I think the sustainability is really something which is truly pre-competitive, where we can exchange ideas and ways of working jointly together. So it's really nice to see that we have all these investments and also different partners and companies to discuss with in our area. For sustainability within AstraZeneca, we rely on 3 pillars. So access to healthcare, which you have in pinkish there. And you have seen one evidence of our vision and strive here in our work with the vaccine, where we had the AstraZeneca vaccine, which has now been delivered to 180 different countries. And we estimate that we have saved 2.6 million lives through our vaccine program. And it's also very important for the company that we are truly transparent with what we do, how we produce our medications and medicines, and as well as how we run our clinical trials and everything there. So the second pillar is the ethics and transparency, which we rely on for the sustainability. And the third is maybe where we're mostly here for today, is about our environmental protection, where we have a very high ambition around ambition zero carbon, which I come back to, product sustainability and natural resource preservation. So talking very briefly about the ambition zero carbon, we have now by 2026, we will have been reduced all our carbon emission from the operations, which I talked about. Our site is really in the forefront here, but over globally, we will be down to having reduced this by 98%. And by 2030, we have our vision saying we're going to become carbon negative. And that is that we, by 2030, we will have been reducing all our greenhouse gas emission by 50% versus the 2019 target level. And the rest we will have offset because we're investing a lot of money into also new reforestation and other programs. And we have been one of the first 7 companies who's been part of this new science-based net zero corporation. So this is an external benchmarking and organization who sets the science-based target index, and which we have presented our strategy and got it approved. So now we're working with that, and they will have external audits on our journeys towards reaching our objectives. So last slide, I just wanted to touch upon a little bit how this fits within the chemistry and how we drive discovery. And I have put this into 4 different pillars here. And the first one is what we talk about avoid. And a lot of the efforts we're putting in today also to drive our discovery work and moving forward to be more productive and being more efficient is to do more predictions. And we have a lot of efforts into doing AI and prediction of design of new molecules, as well as design of synthetic routes to access these molecules. And that will really reduce our waste in a way of not making compounds that we don't think will be as productive. So be able to predict this more efficiently would help us to moving faster and faster into the right molecule space, which we need for our different projects. Another important discovery is how we automate and also run high throughput experimentations. You will see some examples likely later on today where we can more rapidly optimize the way we make our molecules, as well as optimize the synthetic routes to them. So we can really reduce waste products, reduce the functional, or I should say protection groups, etc., and have more efficient synthesis. And this we have also miniaturized, so we get this down. We can do this now in plate formats in microliter scale, and also recently shown that we can do this on nanoliters volumes. And the third pillar here is around late-stage functionalization, where we will also see Professor Ackermann later on talking about where we see that there is an advantage in following the new trends in synthesis, where we can do CH activation and thereby functionalize more advanced drug-like molecule in the second last step, rather than starting the synthesis early on and then make all compounds in several steps. So this will also reduce our chemicals we need to use and more rapidly expand our understanding of structure, activity, relationships, and properties of the molecules. And the third I'd also like to share with you today is around our engagement with the Green Labs. That's an external company or organization that we work with. So we have by today now Green Lab certified all our chemistry labs here at site. And that's a continuous work we do. So we recertify every second year and try to improve further and further how we can be more mindful of ways of working in the labs and so on. And what I want to leave you with them 2 different messages here is, first of all of what I've shared on this slide is driving our business to be more efficient. We save money, we save time, etc. And I really want you to feel that all this, which you can think of as an industry business direction, it goes hand in hand with driving a more sustainable way of working. I really truly believe that it's not contradicting. This is going in parallel. And then the second message I'd like to leave you with is we see this completely pre-competitive. So the more we can work together and sharing ideas how we work on green chemistry and a more sustainable way of working across the industry and with other partners, the faster we will move to a better place to be. So by that, thanks a lot for joining us and have a good afternoon.
Yes, my name is Lennart Bergström. I'm the co-chair of this symposium together with Ulrika Edlund. And I'm going to introduce Paul Anastas. Unfortunately, Paul was unable to travel due to a recent COVID infection, so he will give his talk remotely here. And if you could put up Paul on here, he should be in the waiting room. Great. Hi, Paul. Good to see you.
Thank you.
Paul Anastas, I'm sure that many of you are aware of his achievements, but just a brief introduction here. So Paul Anastas holds the Heinz Chair in Chemistry for the Environment at Yale University, and is also the founding director of the Yale Center for Green Chemistry and Green Engineering. He's commonly referred to as the father of green chemistry, has authored 14 books and numerous, also formed numerous companies in the field of green and sustainable chemistry. And he's also been awarded a number of prizes. Last year it was the Volvo Environmental Prize. 2020 was awarded the King Carl Gustaf XVI Professorship Prize, Stockholm University, and the Royal Society of Chemistry GA's Medal, the Heinz Award, and most recently the 2022 Hoffman Medal. Paul, I'm looking forward to your talk, please.
Well, thank you very much for that kind introduction. Please, if the audience could wave if you can hear me all right. I'm not seeing any waving. There we go. All right. Well, wonderful. So first, let me say thank you for the kind invitation. Let me certainly apologize for not being there in person. So as Leonard mentioned, I got hit fairly hard with the COVID virus. I want to make sure that any of you who think that it's not a big thing, please take it seriously. The last 3 weeks, I'm actually still living in my library right now, and I mentioned earlier about vaccines. There's no greater thankful person in the world than me for vaccines. But if I do occasionally cough when I'm speaking, please forgive me. So I'm going to try to share my slides. Let me see if this goes smoothly. And we will share this. We will go like this. And I'm going to assume, unless I hear differently, that's working. So what I'm going to, I'd like to start off with, and I'm going to try to leave time for at least a couple of questions if I can. And that is, I would like to start by telling you what I'm not going to talk to you about today. I am not going to spend a lot of time talking about my own work going on in my own research group. I'm not going to tell you about my Center for Green Chemistry and Green Engineering that cuts across a number of schools at Yale. Not going to tell you about how we work with everything from the School of Medicine, Management, Law, and Divinity in order to implement the work we do. I'm not going to spend any time talking about how, yes, we do basic research, but we don't end there. We train the next generation. We catalyze implementation through industrial partnerships and spinning out companies, and we raise awareness through various events and conferences. Certainly not going to talk to you about how we use density functional theory and other quantum models to come up with de novo design rules for molecular design for reduced hazards, because if you want to, you can certainly read these papers. Not going to talk about how we're doing catalysis of all types. Yes, we use catalysis and electrocatalysis to oxidize water, to reduce CO2, to carry out other types of transformations. Not going to talk about how we utilize lignin or how we transform many different forms of biomass, because once again, you can read all about it just by calling up a journal. Won't spend any time talking about the new companies that we've launched from this technology, such as P2 Science, Catalytic Innovations, or the Air Company, which has made the first carbon negative vodka, and of which the first bottle of carbon negative vodka went to the king. Or talk to you about another company that we spun out called Kenya. And I'm finally not going to talk to you about how we engage globally with the United Nations on a global green chemistry initiative that's launching into a next phase, a rather large $12 million way of raising awareness about green chemistry. So why am I not going to talk to you about all of those things? Because I want to talk to you about something that I consider to be very important and much more important than my own work. And I want to, in the process of doing that, tell you why my research group and my center is working on all of those things. By the end of this talk, hopefully you'll understand why we do what we do in green chemistry. Now, I'm going to start off by saying two things. One, I love chemistry. And two, I am proud to be a chemist. So I want to emphasize that because through the course of this talk, I'm going to be saying that we need to change things. We need to change how we act, how we do our chemistry, and how we need to think about our chemistry. So I'm going to ask a favor of the audience. I'm going to ask you for just 10 seconds to not move, to stay still. So for 10 seconds, you can blink your eyes, your heart will beat, you'll breathe and things like that. But if you would stay still for 10 seconds, starting now, then I want you just to stay still. 4, 3, 2, 1. Okay. Did anybody move? If you moved, raise your hand. Okay, good. I don't think, I can't see from this camera angle, but I'm going to assume that most of you didn't move, right? You didn't move? I'm going to suggest, yeah, you did move. So you say, you just told me you didn't move. Say that's true, but I'm going to say that all of us moved 4 kilometers in that 10 seconds because of the rotation of the Earth on its axis. That's true. We all said we didn't move. We said that was true. We all moved 4 kilometers. I'm going to say that we moved 300 kilometers in that 10 seconds because of the Earth rotating around the sun. That's true. It's just as true as you didn't move. It's just as true as you moved 4 kilometers. It's true that you moved 200 kilometers. I'm going to suggest that we all moved 22,000 kilometers in that 10 seconds because of the solar system moving through the galaxy. That's true. So why do I mention that? Because in the course of this talk, I want to talk about a few things that are true. And we have to recognize that it was true that you didn't move. It was true that you moved 22,000 kilometers. And the answer of the question is relative to what? What is the context? What is the perspective? And so what I'd like to do is bring some new perspective to what we take as true. Because if we're going to act differently and move towards sustainability, I'm going to suggest we need to think differently. So as a person who loves chemistry and a proud chemist, I am going to say something that's true. The chemical enterprise is genuinely astounding. It's genuinely astounding that manufacturing is becoming more and more efficient, that the petrochemical industry is nothing short of a technological miracle. When you start thinking about the material and energy efficiencies of petroleum refining, for instance, it is a technological miracle of efficiency. I'm going to say that it's astounding because we have materials, we've developed materials that are predictable, stable, durable, whether it is on our spacecraft that such as Voyager, who's out there outside the solar system for 45 years when it was expected to last for 5, whether it's Alvin at the bottom of the ocean, these materials are predictable, stable, and durable. They're resistant to big disturbances, whether it's the thermal shields re-entering the atmosphere, whether it's bulletproof vests and Kevlar, stable and resistant to disturbances. Our reductionist understanding of the universe allows us to deeply understand the fundamentals of our science and build these materials. We're working with some of the most advanced knowledge and bringing it to our chemistry and our materials. And we have high purity materials, 99%, 99.999. We know whether it's in the pharmaceutical industry, whether it's in the electronics industry or beyond, we can attain high purity. And the performance of our chemicals and our materials have excellent functional performance, whether it's hurricane-resistant housing and building materials, such as that house where nothing else was left standing, or being at the bottom of the earth in Antarctica, being able to survive because of the materials that have such high, excellent functional performance. Okay, all of these things I'm going to suggest are true. Some of you have seen the movie The Matrix. You want the red pill or the blue pill? Blue pill, you can keep on believing what you want to believe, or the red pill is the whole truth. So I'm going to take the speaker's prerogative and assume you said, we want the red pill. So here we go. What we produced. The largest product of the chemical industry is carbon dioxide. It's not what I didn't say what we intended to produce. That's I'm saying what we desire to produce. The largest product of the chemical industry is carbon dioxide by far. Perhaps the largest product of the pharmaceutical industry might be waste solvent. The way we transport people and things is changing our climate for centuries. The various hundreds and hundreds of chemical products that we've made are bioaccumulating in our blood in our bodies. And this shows some of the time frames for which those just passed through mother's milk will stay in our bodies. The way that we grow our food, the agricultural chemicals that we use, are damaging the water sources necessary for life. When we talk about flaring natural gas, it's about 20% in the United States. And if we look at the global waste from flaring, we're talking that if the CO2 emissions just from flaring alone, that if it were a nation, it would be comparable with the CO2 emissions of the UK, France, or Italy. And the total volume flared is about, let's call it 35% of the utilization in the EU. When we look at plastics waste in the ocean, we know that there'll be a megaton of fish in the ocean by 2050 and about a megaton of plastic waste in 2050. And we know that the plastic waste is going to outlive all of those fish. So we talked about various truths. We talked about how amazing and genuinely incredible the chemical enterprise is, and then we talked about the whole truth. Well, the truth is that much of our traditional chemistry is not conducive to life, and it needs to change. All right, so let's let's pause, because when we confront this truth, we have to ask ourselves, first, how do we get here? And two, how do we get on a different path? And how do we think differently to act differently? The elephant in the room. It's uncomfortable for all of us to consider that it's possible that the exact things that gave us all of the successes and the victories of the past, our greatest achievements, might be the exact things that are contributing to our demise. So, what do I mean by that? The things that I listed off as being truly astounding are listed here. Efficiency, stability, resistance, knowledge, purity, reductionism, performance. So I'm going to ask the audience, and I can see a little bit of the audience. You know, we all were raised recognizing that efficiency was a good thing. We even heard this morning that we strive for efficiency. Who in the audience would agree efficiency is a good thing? Please raise your hands high so I can see them in the little picture that I have. Right, so we know that efficiency is a good thing. Thank you very much. I'm going to suggest we're wrong. I'm going to suggest that efficiency has no value. And I'd like to prove it to you. So, the reason I know efficiency has no value is because how many people would want an efficient robber or an efficient thief? How many people would want an efficient carcinogen? How many people would want an efficient mass murderer? Right. No one. And why is that? Because efficiency has no value. The value, the nobility, the desirability comes from the act itself. Making a bad thing more efficient doesn't make it a good thing. So why would I spend all this time talking about this? Because so often in our drive for sustainability, we're seeing that we are trying to make fundamentally unsustainable products and processes and manufacturing more efficient, even though they are fundamentally unsustainable. So trying to make an unsustainable product more efficient is a lot like walking north on a southbound train. So efficiency has no value. We need to look at the underlying elements, the character and nature of the materials, energy, and the products and the processes themselves in order for it to be efficient, in order for it to be desirable, I should say. So efficiency, I like to say, will help you do the thing you're doing better, but it will not help you do a better thing. When we talk about something that's stable and static and reliable, we recognize that so many of our products are persistent, bioaccumulative, biomagnifying. We have set the stability and durability of our molecules and our materials as a good thing, but yet we are seeing that that performance aspect alone is something that can come back and cause harm. I often say to my colleagues at Yale, you know, it's fine if we want to pursue immortality. It's not fine if we want to impart that immortality upon the materials and the molecules that we make. So resistant to problems. We find that these types of designs work well right up until they don't, and then there's catastrophic failure. We judge these things through risk analysis. What is the probability of something happening? And if the probability is low enough, we call it to be an acceptable risk. We'll call it a black swan event. Well, right now, I don't know if any of you have noticed, but we are dealing with a flock of black swans, those those low probability but high consequence events that we're seeing, whether it's so-called natural disasters driven by climate change and others. Knowledge. We all know that knowledge is a wonderful thing, right? Well, we also recognize that knowledge is 3 clicks away on the internet. And fragmented, isolated bits of knowledge is only useful when it's transformed to create insight, wisdom, and informed design. Purity. We strive for purity. Why? We strive for purity because purity means control. It means we understand the situation. It means we understand the manufacturing process. It means we can control it, right? Yet we all know that purity comes at an extremely high price. This fact might be a little out of date, but just looking at the distillation columns in the United States that are used to purify large commodity chemicals, they use 3 times the energy of the country of Sweden. Now, maybe that's been decreased a little bit, or Sweden has used more, but it comes at a tremendous cost of material and energy. Reductionism. Reductionism, holding all parameters constant and changing one to understand the world, has allowed us to get an insight into the universe over the past 200 years that allowed us to create the modern world. Reductionism has been astounding at allowing us to understand fundamental laws, basic principles, but yet it's not the way the world works. The world doesn't work in isolation. And one of the greatest quotes from Philip Anderson in a 1972 paper, he talked about how our ability to reduce the world to a set of natural laws in no way implies an ability to apply those laws and recreate the universe. So reductionism is not the same as systems thinking. And finally, performance. A sole focus on primary functional performance, usually at the use stage, has often led to all of the unintended consequences that we've been talking about. Unintended consequences throughout the life cycle, whether it's depleting of feedstocks or end of life. So we look at the pillars that have given us so many of our astounding accomplishments, and we call them into question. And so how do we start thinking differently? Is that we need to throw out this way of thinking? Or is it that we just need to evolve? So I'm going to suggest that evolving the way that we're thinking is where we really need to go. So efficiency, evolving to effectiveness, is nothing more than looking at how nature, natural systems, biological systems work. And we need to recognize that nature is not efficient. Nature is effective, whether it's photosynthesis or leaves falling off the trees, et cetera, et cetera. Our natural systems, or our own physiology, our natural systems are tremendously effective, but not efficient. And when I say effective, all of these come back to being conducive to life. The idea of something being stable and versus stable and static versus dynamic. So nature is dynamic. The world is dynamic. Things are going to change. How do we start thinking about our materials? How do we start thinking about our energy so that they adapt and adjust over time to take on the properties and the character that we need? How do we make our materials and energy more dynamic? We have things that are resistant. We want to try to make it more resilient. Resilient is what integrated complex systems use to survive. Resilience rather than resistant. So knowledge needs to evolve, data into information, information to knowledge, but knowledge needs to evolve into insight and wisdom so it can inform design. And the value of the knowledge is not how deep and specific it is. It's how many interconnections that it makes in order to enable new creativity and new design. Purity. We love purity because it means control. But nature, once again, is not about having storing proteins over here or storing carbohydrates over here. Nature is able to deal with mixtures, extremely complex mixtures and biological formulations, and have all of these taking place. Why do I mention that? Because by looking at nature, we recognize that it's possible, right? And so looking at what's possible and recognizing that the price we pay for feelings of control is extremely high in terms of energy and in our designs. Reductionism. Reductionism needs to shift to our systems thinking, recognizing, as John Muir once said, once you tug on anything, you wind up realizing it's connected to everything else in the universe. And so that is true of our chemistry and understanding what systems chemistry actually means. And finally, performance. In addition to having very specific performance of our materials, whether it's thermal conductivity or insulation or water repellency, whatever that primary performance is, we need to understand that that needs to be coupled with sustainability, that it must accomplish its function without causing problems for humans and the environment. All right. So I'm trying to advance this slide now. We'll see if this works. My slides have stopped advancing. There we go. So what does all of this shift in thinking mean for the chemical enterprise? What this means and what the future might just look like is the future is photons and electrons. For those of you who haven't seen a industrial scale electrolyzer or electrocatalysis, that's what it is on the right. Photons and electrons rather than phosgene, fluorine, and cyanide. The idea that we carry out the vast majority of our transformations using these as drivers is one part of what we believe the future would look like if it were conducive to life. Dynamic rather than static. So when you're asked what color is your car, and you may want to say, well, what color would you like it to be? Because upon stimulus, we can certainly have color changing cars, but it's more than just changing the colors of your car or your shirt. When somebody says, what's the polarity of your solvent? You'll say, what polarity would you like it to be? Upon imparted stimulus, whether it is light or pH or magnetism or electric current, you'll say, what polarity would you like it to be? Because we want to engage in self-separation. When we start talking about how do we separate complex mixtures, it's about how you design dynamism for self-separations. So there's work going on in these areas, but it's a small sliver. Changing our chemistry from static to dynamic is one of the great challenges of the future. Lasting as long as we want things to and no longer. The idea that we want our materials to last millennia is sort of an anathema to being conducive to life. So We want these to last as long as their useful life requires and not a minute longer. And once again, this designed for degradation or triggered self-healing on the other end of the spectrum. This is about challenges of design and how we impart these properties into our materials. Once again, this is going to be necessary. If we're going to talk about a circular economy, then we're going to need to understand how to deal with the energy it takes to either separate those things that we want to continue in the process, or have those other things degrade harmlessly into the environment. Making things from renewables and waste rather than virgin feedstock. The reason that we do so much in the area of bio-based and renewables is because we want to have our carbon be in the natural carbon cycle. We want to make sure that the elements that we use are going to certainly be in the not in the critical rare earth range. And perhaps if we're lucky, decades into the future, people are going to look back at the achievements of the 20th century, sorry, the 21st century, and say, we took our most problematic waste, CO2, and turned it into a source of value. So when we look at what can even now be done for transforming CO2, we know that we can transform CO2 into building materials. And one of the companies that we have spun out is a company called Calcify from Carbon Capture Machine. So turning this into building materials, we developed this carbon negative vodka that I mentioned earlier to capture people's imagination, not to cure climate change, to capture people's imagination so that they could recognize that if you can capture things from the air like CO2 and transform it into a carbon negative vodka, then you certainly can do things with CO2 on a larger scale. And on Friday, this past Friday, there was an announcement that the first commercial sustainable aviation fuel from CO2 was launched by the air company and is being purchased by JetBlue Airlines and Virgin Atlantic Airlines. So I encourage you all to take a look at that. So that's what's going from the bench to commercial scale looks like. Systems thinking will be absolutely crucial. How we understand the interconnections of things, how we understand the drivers. As we see in the bottom left-hand corner, this is this idea of chemical leasing, recognizing that no one ever bought a chemical. No one ever bought a chemical. They bought performance, they bought function. And so if you can provide that function, that service, that performance, and provide it as a service rather than as an object, all of a sudden that changes the incentives. You want to use as little of the material, generate as little of the material as it takes to provide that service, rather than produce as much in order to sell as much as you possibly can. These ideas of systems thinking and systems design will be crucial rather than linear design. I was lucky enough, I guess this past year and a half ago, to publish a paper in Science with my colleagues Julie Zimmerman, Walter Leitner, and Hanno Rütherpel. We were invited to write a paper called Designing for a Green Chemistry Future and Chemistry for Tomorrow's Earth. And I just want to mention one chart in this Earth because it really does capture the transformation that we're talking about. And that is that in order to move from linear to circular, we're going to need to make a few different transitions from fossil to renewable, to reactive persistence and toxic chemicals, to benign chemical reagents and protocols. Catalysts using precious and rare metals to earth abundant catalysts, moving from covalent bonds only to recognizing that nature uses weak force, non-covalent interactions. We want to move to low toxicity and obedient solvents. We want to make sure that our dynamism is in our solvents, in our materials, in our catalysts. And we also want to pivot toward waste utilization rather than waste handling and waste treatment. So I won't go through all of these. I'll recommend you to the paper if you'd like. But I want to just finish by saying that we all dedicate our lives to discoveries in our research. Those discoveries make a different future possible. 2 years ago, we put out a piece in celebration of the periodic table's anniversary, and this is using the periodic table as a metaphor. The periodic table of the elements of green and sustainable chemistry. As you can see, I have it right behind me, and all of the elements in the center of this periodic table are those scientific and technological advances that we all focus most of our time on. The catalysis, the synthetic transformations, the separation, all of that science and technology is right at the heart of the periodic table of green and sustainable chemistry. But it's to the left that are the humanitarian elements. It's the reasons that we do the work we do. It's the food, it's the shelter, it's the providing of water, it's making sure that the medicines exist. All of those are the driving reasons. But to the right on the periodic table is all of those things that are going to turn our discoveries into impact. Those things that are necessary to bring the discoveries and inventions and innovations to scale. So will it be new conceptual frameworks that we think about, like biomimicry and circular economy? Yes. Will it be new economics and new metrics in order to drive and enable these things going to scale? Yes. Will it be new policies and regulation? Will it be new tools for understanding? These approaches, these conceptual frameworks are crucial to having our discoveries and our innovations go to scale, as we aspire to the far right, which is the noble elements of our visions for a better future over the horizon. The center, this is green chemistry. This is what chemists and engineers do. This is sustainable chemistry. This is what everyone should do. Our economists, our policymakers, our teachers, our mathematicians, our marketers, our communicators. And this is how green and sustainable chemistry fit together. Green chemistry is chemistry. Sustainable chemistry allows green chemistry to make an impact on the world through all of these other disciplines. And just like we talked about valence in terms of valence electrons, valence means making connections and creating new entities that bring about transformations. This is a book that came out. If you want to take a picture of this, this is a free book. It's a small gift to say thank you for the invitation. It goes through each element one by one and explains how they all interconnect. So please feel free to go to this website and download it if you would like. And I just finished by saying this quote from Mahatma Gandhi. The difference between what we do and what we are capable of doing would more than suffice to solve most of the world's problems. Thank you, and if we have a couple minutes, I'll be happy to answer questions. Much appreciated.
Thank you very much, Paul, for an excellent talk. Yes, we do have a few minutes, so the floor is open for questions or comments.
I tried to be provocative.
Anyone who wants to go first? All right, Paul, let me start then. So I was actually teaching green and sustainable chemistry just a couple of weeks ago for a group of about 20 very ambitious and interested students, and got a lot of questions. And one of the questions was about the quite often when you develop a process and apply the 12 principles of green chemistry, sometimes you have a benefit in one of the principles and maybe an offset in the other one. And the question was how to judge this. Are there any sort of rules also within the green chemistry concept, or does this goes back to the sort of the judgment as a chemist?
Oh, so it's interesting because for years people would be saying, oh, well, there's no green chemistry, there's only greener chemistry. And, and I laugh and say, the reason that, I introduced the term green chemistry is because it necessarily implies a goal of perfection. Perfection can never be reached. And that's kind of the point. You want to guarantee continuous improvement. And it's only by striving for that ideal that we guarantee continuous improvement. So when you wind up in the case that you just suggested, where there are trade-offs, it means your design challenges aren't done. What it means is that there are very few, if any, inherent challenges, inherent trade-offs. They're almost always trade-offs of circumstance, okay? So if they're circumstantial, those can be addressed through appropriate design. But if people run into challenges where there are inherent conflicts between the principles, that's something that I'd want to know about.
Thank you. Any questions on the web? No? Oh, it seems that you have been extremely clear here, Paul. I think, we have one. Okay, very good.
Thank you. Do you hear me?
I can hear you.
Hello. Now you hear me better. My name is Lotta. I work for a company called Per-Olof. We do a lot of synthetic chemistry. I'm not a practicing chemist anymore, but I think this was extremely inspiring. So thank you very much.
Thank you.
However, I do see some ground to be covered between the future that you present and the chemistry that we have out there in a lot of production sites today. How do you see that we can get momentum to have a real transformation according to these principles that you describe?
Yes, so I completely agree. There needs to be a lot of change, and I do believe that the only way that you change what you do is you change how you think. What is interesting to me is seeing, now this is a generalization. When I see startup companies, smaller companies, they are often talking about, when it comes to sustainability, what they can do, what they're capable of doing, the new things that they're able to achieve. When I hear the largest companies, too often I'm hearing, well, what we can't do, what we're limited from doing. We have established, factories, pipe in the ground, embedded investment. So these are all reflections of our current ways of thinking. So part of the reason why I asked everybody to sit still and then talk about how fast we were moving is because those different truths exist simultaneously. And we talk about what is the context. When we start talking about our economic models that have made and informed decisions that have brought us to an unsustainable world, those can be true in their own context. When we start looking at alternative economic models, they can be equally true, but will drive very different decision making. What do I mean by that? I mean that if we say, oh, using our current economic models, this is the most economically beneficial, we'll have to say to whom and for whom. What is the context? What is the relevance, in the same way as we're traveling through space, right? When we use our new economic models, we'll say, okay, this drives different decision making. This would require that we actually use different feedstocks, different separation methods, different synthetic methods. Why? Because it will allow us to preserve the exact resources, the exact, I'll call it natural wealth, that allowed us to achieve our greatest successes in the first place, rather than destroying those resources. So it's not moving from, one thing that's true to another thing that's false. It's about changing our perspective in the way that we choose which models we're going to obey.
I think with that closing words, we have to close this. And thank you again, Paul.
Thank you. Thank you. Be well.
So we're going to continue now with the poster pitches. We have 7 eager PhD students and postdocs to have 1 minute each to present their posters. So let's start with the first one. And you present yourself and your work. Yes. Please.
Can you hear me? No? Hello, I'm Pedro Tortajada, a PhD student from Stockholm University. Our work focuses on improving the environmental impact of alkene hydrogenations. By performing the reaction in electrochemical conditions, we are able to access the transformation using a sulfuric acid solution as well as a nickel foam as catalyst. The method shows a good performance for a variety of substrates, as well as good recyclability properties. Furthermore, prospective LCA and hazard assessments will provide us information about the safety and environmental performance of this method and compare it with the classical palladium on carbon approach. So if you have further doubts, we can have a further discussion. I will be pleased for that.
Thank you.
Hi everyone, I'm Haibo from Stockholm University. In this work, we have developed a sustainable methodology for the synthesis of diverse amine by using this cellulose supported heterogeneous non-copper catalyst. So this will be the first example of synthesizing amine by using heterogeneous catalysis, and we obtained wide scope, and especially for this di and tri substitute amine, we can obtain excellent caloric transfer. And furthermore, we apply this method for the total synthesis of 5 natural products. And if you have interest in this work, we can have more discussion outside and in front of the post. Thank you.
Hello. So we are working on the development of high content screening protocols, to detect sublethal effects after chemical exposure on Daphnia, which is this weird creature over here. And with this, we hopefully have a new, faster and accurate tool to assess industrial chemicals. And how are we doing this? So we are basically exposing Daphnia to chemicals, and then we are doing a systematic treatment with fluorescent compounds. And these compounds would allow us to visualize different toxicological endpoints. And with this information later on, we are going to see what implications this exposure have in growth and reproductions of these organisms. So if you are interested, please, I'm going to be happy to have a discussion with you. Come and see my poster. We're the Daphnia people. So thank you.
Hello, I'm Cedric, PhD student at Stockholm University. And this probably looks for most of you like something from a science fiction movie, but it's actually how we see Daphnia under the microscope. Here we applied several fluorescence stains that allow us to monitor the health status or biochemical effects in the Daphnia. The advantages of this method is not only that we can see a toxic effect, but also where and when it appears. Since this is an image-based method, we can literally follow the toxic effect by eye. But we can also use the advantage of a highly sensitive camera, which is able to see a lot more than we can do. So if you're interested more into how this molecular staining is working, how we use this method to assess toxic effects in chemicals, or how we put these basic pictures into numbers, you're very welcome to visit me at my poster. Thank you.
We are being exposed to phthalates, bisphenols, PFASs, as an example, and we know this because we are measuring these compounds in our surroundings. But what about the other hundreds of thousands of chemicals that we use and don't even look for in our environment? So we are working on a hazard screening workflow, and the hazards we are screening for, they are organic pollutants in our waters. And we are not only targeting the pollutants we already know exist in the environment, but we are also looking for the unknowns. So if you're interested in discussing or learning more about how we collect and work with this big data, please come have a chat with me by the poster.
Hi, my name is Zia. I'm a postdoc in computational chemistry from Cytiva in close collaboration with RISE. So in Cytiva, we are trying to identify the sustainable alternatives to the chemicals we are using right now. So for that, in Cytiva, for the past few years, we have already developed a toolbox. But the challenge here is that for some toxicity data, we cannot find anything. So here I came in to develop prediction tools to fill in these data gaps. And here in Mistra SafeChem Workbench 3, there are more modelers developing wonderful models to predict toxicity data. I'm not going to tell you a lot because right after the coffee break, Ian from RISE are going to tell you more from his speech. But if you are interested in how we develop this whole toolbox in Cytiva, you're welcome to my poster during a coffee break. Thank you.
Hi, everyone. I'm Kerstin, a PhD student from the Technical University of Denmark. When we want to do life cycle toxicity assessment, we need a lot of data for all kinds of chemical and material properties. Finding these data can be difficult and very time consuming. So why don't we just build machine learning models for all of these? Well, there are two challenges. First of all, machine learning might not be the most suitable approach to address these parameters. And secondly, there are a lot of them. And at the same time, they are not all equally important for the model results. We prioritized all input parameters to our toxicity assessment models for their suitability and for the urgency to develop machine learning-based prediction tools. So if you're at the conference, do take the chance and take a sneak peek at our unpublished results. Thank you.
Yes, thank you very much for excellent poster pitches. Stick to the time also. There is now coffee outside. No need to use the machines. There is coffee served separately. And please be back here at 2:30 to keep the time. And the posters, you will find them also to the right.
Okay, everybody, thank you for coming back. Participants on Zoom, welcome back. People in the house, welcome back. It's time to resume this symposium on green chemistry. All right, as I said early on today, Mistra SafeChem has quite an ambitious vision, and we have a really strong team to address the challenges. So just before coffee, we heard some of the postdocs and PhDs within the program elegantly present their work. And now it's time to move on to the principal investigators. We have 3 principal investigators that will tell a little bit about their ongoing work and the projects that they lead on building and utilizing a toolbox for safe and sustainable chemistry. First out is Professor Ian Cotgreave. You're at Karolinska Institute and Rice Research Institutes of Sweden. Please, Ian.
Does everybody hear me okay? Center stage. Went to scene school once upon a time. We'll do a little dance. Okay, fantastic talk. We just heard about the big picture, the universe revolving around us and we revolving in the universe. Now I'm going to bring us down to town a little bit with some tools that we need to use. And I'm going to focus on in silico tools. So everything that's done inside a computer these days to try and predict properties that might be less than desirable, let's put it that way. So minimizing hazards and risks also, obviously. If we're not exposed to things, hazards are not risks, but we generally get exposed to hazards as well, and they become a risk. And some of the tools I'm going to show you are useful. We've developed them to be used within the SafeChem program, and they are multifactorial. There are different tracks of development, traditional machine learning techniques I'll show you, and also artificial intelligence-based, non-descriptor-based methods as well. So next slide, please. Can I press that or? Oh, it's one up there, is there? Okay, good. High tech. Here we go. So obviously, this is the general picture we've seen from Mistra SafeChem before. And I just put a few arrows on here saying where these kinds of techniques can be used in this cyclic process. So safe by design can be achieved using in silico-based tools with multiple input areas. So it's not just one area in the cycle that we can use these techniques. And of course, they're related to some of these principles of green chemistry, as we've heard, where obviously designing syntheses to use and generate substrates with little or no toxicity, obviously, is very important, either to humans and the environment, that are fully effective and have little or no toxicity. And also to design chemical products to break down to innocuous substances, as we heard from Paul. This is very important. You need to almost plan in the destructibility, like my Volkswagen Passat is just destroying itself after 10 years. All the bits and pieces are failing on me, but it's done by design in order to be innocuous. That was a joke, by the way. Some people got it. Obviously, identifying hazards in the safe design by design paradigm. And I'm standing in the building, which used to be my I was employed at AstraZeneca, where safe by design, obviously in pharmaceutical agents, is extremely important. If you can do that early on, you really get a pharmaceutical agent which is highly potent, which minimizes the risks. And so a lot of the technologies, the in silico-based tools we were developing in the pharma industry 20, 25 years ago, and they are now finding application in what we called in those days front-loading knowledge. In other words, putting knowledge early on into design and synthesis. And obviously you can do this in several ways. One way is to chase things around the literature, the old school way, and there is a lot of data out there. So you can either provide hazard information by actually looking in the literature, in the ECHA databases, in the EMEA databases, and so on and so forth. Or you may have data gaps because they're new chemists, new chemistry, new processes. You might have to generate some data. Generally, though, you're in a situation where you can use both. And so both are relevant here. But clearly, generating new data is important. And how do you do that? Well, traditionally, of course, toxicological data has been generated in vivo, in animals, plenty of time, and also using cell culture technologies and so on and so forth. That's the world I grew up in from the beginning. But of course, these are time consuming. There's a lot of pressure on the use of animals in risk assessment. The Directive 1063, which came 12 years ago now, really pointed out that we need to take care of replacing, where we can, animal experimentation. It's costly, takes time. There are some areas where it's still prescribed. Obviously, you're developing a drug, you need to go into an intact organism. No reductionism there whatsoever is tolerated. But of course, in the world we live in today, rapidly we are providing the ability to do this by predictive modeling in computers. And so this area is now becoming very attractive and tractable for use in projects like the SafeChem project. They generally are, obviously they're trained on experimental data, so they are pretty well informed. They're often cheap and fast. Zia will tell you, not really fast. They have to be built, right? That takes time. Relatively easy to use if you're well trained and if you're taught how to do that. And if you have a good interface where input and output comes out and it's understandable to you and explained. There are also high quality models that are possible. And today, traditional machine learning is one thing, but also we're using, as I said, descriptor free approaches using artificial intelligence, which actually teaches itself the best way to make the model and the predictions. And interestingly, importantly, regulatory authorities are becoming much more at ease with the use of computational data in their decision making. And so there are many reasons why in silico modeling would be a good proposal here in the toolbox. And of course, they're used in various scenarios. I'll show you one scenario where alternatives prediction is important. So if you are trying to replace one thing with another, how do you make that decision? How do you weigh the things up? What gives you the weight of evidence? Of course, there is the situation of de novo profiling, if you like, when you're starting from scratch and you're having to design a process with chemicals that you've never worked before. You would like to know something about their properties from the word go. They're not part of an alternatives scenario. And of course, several of the cases within SafeChem they are dealing with these situations in different ways. My colleagues will show you more selectively how these particular techniques are being applied in these case studies. I'm going to keep away from the case studies, just talk about the basic techniques. This is traditional machine learning flow process. Obviously, you start the left-hand side with some data sets where chemical structures are converted into a string where you can put them into a computer. The computer will understand that that's a carbon, that's next to a nitrogen, that's next to an oxygen. And they're converted into descriptors. And the descriptors are various things that you put physicality or chemical structure specific descriptors. They go into the machine learning modeling and they produce a model. And that model has a large degree of validation in it. And at the bottom, you just see that process essentially working from a computational aspect. And I don't really want to point out very much more, but I will come back to the use of neural networking in AI-based predictions. This is traditional machine learning. And of course, the important thing is here at the bottom, when you put in a new chemical challenge, you want to ask the model, is that likely to be hazardous in a particular way? Yes or no? And the question today is also, how sure are you of that? How unsure are you of that prediction as well? Because that's what the regulators are interested in. But this is essentially a standard machine learning approach. And the toolbox that we built, and we built it with multiplicity and overlapping on purpose, because we want to have models that are built in different ways that address the same hazard, because you want to have compliance between, it's a convergent process. But essentially we are dealing with human toxicologically relevant endpoints, endocrine disruption, ecotoxicologically relevant products, We also have some physical, chemical, and pharmacokinetic modeling that is important to understand how in the body, the human body, compounds are disposed of. And also we're developing other models to fit into the toolbox. And I'll just cover a couple of them in the following 5-6 minutes. Obviously, the alternatives assessment route is where we're often in our chemical process. So you really want to make informed decisions about providing an alternative to this one here. Should it be A or B? You don't know. It's a seesaw based on knowledge and input, trying to choose the correct alternative, which is often where we are. And Zia showed quite convincingly in one poster the work that he's been doing, if you read it outside, to look at these are some very important hazards, of course, from a chemical safety perspective. These are the so-called CMR hazards. So all the regulators are really interested in these. For instance, carcinogenicity and mutagenicity have no threshold in regulation. They are, if you have them in your box, in your chemical, in your structure, there is no tolerance. There is no threshold of the biological effect. And so clearly you would like to steer away from a structure that has those particular potential hazards. Reprotox is almost at that particular level. And one of the areas that Zia is focusing on is skin sensitization due to bioreactivity. In other words, generating reactive intermediates in the body, which become biologically hazardous, in this case by the immune system. And of course, Cytiva are interested in this because they use a lot of reactive chemicals in their chemical processes, of course. And so basically he's designed a whole bunch of tools with varying degrees of accuracy, and these are just measures of accuracy. So 100% hit rate means you have 100% predictability. It is a carcinogen, no doubt, 100%. But all times you'll find that the model, when you train it and then address with unknown chemicals or a group of chemicals that you are validating the method with, you will never get 100%. So if you're looking at a predictive model that has 80 to 90% predictivity, that's pretty good in computational terms, I would say. And you're going to see those numbers appearing several times in the toolbox descriptions that I'm coming through. And of course, he's already shown you this picture. And essentially what it's saying is you're trying to fill gaps in some sort of a logical matrix where decisions are being made. And those decisions can be based on information around hazard, information about process development, energy, economy, whatever you like, whatever company you're in, whatever situation you're in. But clearly, these tools can fill gaps where you perhaps don't have information coming from the literature. And the tools in the interface should be secure, data should be secure, it should be accurate, have broad coverage. In other words, it's not just specific for a particular area of chemistry, which is ideal, but in practice rather difficult. So tools need to tell you whether you're in domain or out of domain. If you're out of domain, there is no prediction there. Sorry. Then you have to train it wider, use a wider training set of chemicals. Easy to use and fast. And the toolbox that Zia is developing has all of these characteristics in an interface. One of the interesting things that we're also trying to introduce here is that, again, in the OECD guidelines TG 442, which addresses skin sensitization, and the adverse outcome pathway number 40, which is a way of organizing the process, the mechanism in the OECD's AOP wiki. The first thing that has to happen is that a molecule has to become chemically reactive to bind to a protein in the human body, to become antigenic and to elicit immune response. And this step has been missing from computational toolboxes, combining that step with the actual hazard of developing skin, a sensitization event in the skin. And so Zia is trying to work with combining prediction of metabolic activation to a reactive intermediate with the end point, the end game, which is being allergic to something through the skin contact. And also being able to do that by predicting which part of the molecule is actually the reactive center. When it goes into the body, it's turned into something by an enzyme. This is the reactive portion of the molecule. So it's tracking, so to say, what the body is doing to it. So this is research that Zia is doing, trying to, and nobody's ever done this before. It's being done within the Mistra SafeChem program by Zia and colleagues at RISE. And of course, where do you get your data from? This slide just illustrates essentially that your models are only good as the data you put in them. And obviously where you get that from and your access to public data is very important. AZ has its own data banks with its own chemistries. We don't have access to those, but we do have access to, for instance, data that has been used to train things in the OECD toolbox, the QSAR toolbox. There are other hubs around, and we have our own data in-house. We've collected chemical hazard data from many thousands of compounds for different endpoints. And clearly this is very important. In a second area of modeling, which is more using non-descriptor based techniques. In other words, you don't have to describe the structure of the molecule in a particular way. You use what's called neural networking to describe, collapse down the structure into vectors and put them into an AI. And in this case, I'll leave it at that. There's a lot of computational work in there to build a model that you can actually, is much more flexible. It can be used to track back what was the toxicophore, for instance, what was the part of the molecule that was causing the hazard. And I'll show you that in a second. And there's a couple of examples of the use here. For instance, we built an endocrine disruptor toolbox, which is very important for our environmental perspective and a human health perspective, looking at these various types of receptors. They're important that the molecular initiating event, the first thing that happens in the adverse outcome, binding to these receptors, triggering the responses, granting agonism or antagonism of these receptors. And now you're starting to look at some accuracies up here. So 1 is 100% prediction. So anything that gets close to 80, 90% here is extremely good. And the models that are being developed are learning all the time. This is iterative. So these AI-based models are teaching themselves the best way in which to model the prediction in hand. I don't want to go through any more of these. I'll just show you a few more. We have a bunch of human endpoints that are really important here, but you'll see that the accuracies here are a little bit lower because these are very complex processes we're looking here. It's not just looking at the binding to a receptor. This is looking at liver toxicity. There are 28 mechanisms to cause steatosis in the liver. A bit over the top there, but there are at least 5 or 6 main mechanisms of causing steatosis, fatty liver, which is a disease we don't have. But clearly, that's just one area of liver toxicity. So all the data in there is clustered together around something called liver toxicity. And obviously, this is a complex process, and the simple correlation is not that easy to draw. But it's surprising how close you get, for instance, with mutagenicity here, which is a clearly important process that we don't want to be seeing elicited from our chemistries. So this is an area that we're working on as well. And also, toxicologically relevant endpoints. So this is just looking at a few parameters that are really important from an ecotoxicological perspective. Biodegradation, bioconcentration, persistence. We're building models here, but we haven't looked at Daphnia yet, lighting up in a model. So, but we'll get to that with our collaborations. So this is a bunch of AI-based models that we are using also in parallel to understand hazard. And the reason why we're doing this in parallel is because if you have models that overlap, you can use them to ask the same question. And if you get consensus, that's a stronger observation. They were developed in a different way. And this is just the interface that we use. What we're now using also is these models are being questioned about their fidelity in insecurity of prediction. So as I said, by combining the observations made by many different models, you can start to see how secure or insecure am I in my prediction? And that is a question that's very important if you're going to deal with a regulator. Today, it's more about insecurity of observation than security of observation. Those of you who interacted with regulators, you'll know that. The second thing I mentioned is that these AI-based tools can be used to look, if you're looking down from the left-hand side to the right-hand side, from the events, the prediction of the hazard, once you find the hazard, you can look in the other direction and look and see what part of the molecule actually is resulting in that hazard. So this is toxicophore prediction in the same way as we use pharmacophore prediction in pharmacology. So this is something that we're working on. There's an interface in there now in the toolbox. And of course, what do you use these data for? Well, obviously they're used, or these methods, they're used in our case studies. We have an H&M case study on silicones where we've applied the tools. We also have a Perstorp case study where we're applying the tools, and a couple of ones with arrows here. Well, I think we'll hear a little bit from PO and AGI later on. And there are obviously more to come in the process as we develop. This toolbox is open to all. It's open to all to discuss how to use it. And it's open to all at any time to come in and give us a query or a question. We are also helping out with interpreting the data. And of course, the data goes into the LCA-based methods that we're using as well, and they become relevant there. So that's me, done. Whistle-stop tour around computational toxicology. These are colleagues that I'd like to thank. So it's Ulf Narinda, Swapnil and Zia in the room. Zia has a poster, but Swapnil, put your hand up. Right, there is. Anybody who wants to talk about AI-based technology, he's the man. So I thank you for your attention.
Thank you, Jan. I believe we have time for one quick question, if any. There's nothing online, but we have a question over there.
Peter,
Peter?
Yeah.
Peter?
I could probably hear you.
Oh, but people on Zoom cannot, so please wait for the microphone.
Oh, okay, on Zoom, yeah.
Thanks, Ian. Very exciting. Peter van Kempen from DTU in Denmark. Quick question. Seeing all this, that you have this wealth of AI and machine learning based tools at hand, and then we use it to predict things that others have tried to predict earlier, hazard things like bioconcentration, which is just one very specific point, acute ecotoxicity on a specific species. Whereas we all talk about mixtures that are out there. We talk about products that integrate full life cycles and pathways. Where would you really see AI could be used in a bigger picture than running after specific endpoints? I see a huge ambition that we could build within Mistra SafeChem in the future to be more to make things more complex, to predict things that are intrinsically more complex and capture that, because the tools that we currently use cannot do that.
Yeah, I agree with you, Peter. And the neural networking that we're using, so deep learning through neural networking, the nodes can be anything. They can be relationships between anything in multi-dimensions. And so I agree, we should perhaps start to think about using these deep neural network type methods in integrating data from different points and different questions coming in. The mixtures thing is a standing order one as well, obviously, from hazard perspective, but they are regulated singularly, unfortunately, today. But we'll talk about that more. But you're right, you're absolutely right.
Okay, thank you so much, Ian. Our next Mistra SafeChem speaker is Per-Olof Syrén, Associate Professor at KTH Royal Institute of Technology. And Per-Olof, you will talk about enzymes.
Yes, can you hear me? Cool. Hi, everyone. Great. So I'm going to talk about what we do with enzymes in Mistra SafeChem. So this you already know, it was already mentioned, but what I think is very cool with the Mistra SafeChem program is that it's a close collaboration between academia, industry, and research institutes to tackle grand challenges in green chemistry. And I think we learn a lot from each other. So I am part of VP4 of Mistra SafeChem, and these are the PIs of VP4, my co-PIs. So that you know, I guess some of you know these people, and those in the program, of course, you know this. But I think it's nice to say. So I love enzymes, actually, you know, and I will come to why soon. So my research actually is at the focus of protein and enzyme technology. And we use enzyme design and protein design to actually develop better catalysts. That's what I'm going to focus on today. But also the techniques that we use are also actually employed. We use them, for example, in biopharmaceutical research to make better drugs, etc. But I'm not going to speak about that. I'm going to speak about the catalysis today. Okay, so what's an enzyme? Well, an enzyme is like any chemical catalyst. It's working by lowering the Gibbs free energy of activation for the rate-limiting step. So that's the key. And it's basically a highly structured protein with an active site that is highly prone to engineering. And I'll come back to that soon. So enzymes are actually quite efficient. This table here I compiled some data from literature. It tells you what happens if we wouldn't have an enzyme in processes that are relevant for life, such as energy metabolism. So if we now look, for example, on this is an important reaction for example, energy. So if we wouldn't have had enzymes, the half-life for this reaction would be about 1 million years. So that means that having a cell that would divide every million year, that would be a very slow life. So it doesn't work. So enzymes are key for life. They speed up the reaction by lowering the Gibbs free energy of activation. Okay, so the idea here is that enzymes, as enzymes are efficient, we should use them in industry and in catalysis. This is actually inspired from Paul's talk, you know, just want to show this again. We are talking about the 12 principles of green chemistry. And I think enzymes fit nicely and snugly in here because we can do specific catalysis such as regio, stereo, enantio, specific catalysis under mild conditions in water as solvent. So that's green chemistry to me. One example actually is, this is one, well, maybe you know about this story, but it's about generation of taxadiene or Taxol, which is actually the active drug. Taxol or taxane, actually, taxadiene was actually traditionally extracted from the taxus tree. So to make one dose of this anti-cancer drug, you needed to take down one tree that's 100 years old. So that's not sustainable. Then, of course, you can make chemistry and synthesize this drug. This is not the latest technique. There are now more elegant routes to taxadiene and Taxol. But I just want to show this. It can be quite complex. And what I'm saying here is that we can learn from nature. So actually nature makes this molecule. And it is actually possible to make catalysis that looks like this by employing enzyme catalysis. In this case, it's an enzyme called a terpene cyclase that makes this multicyclic chiral scaffold in one step in water, which is a quite cool reaction. It's carbocationic base. I'm not going to talk about the mechanism because I don't think I have time. I made this slide. It's kind of myths and facts about enzymes in industry. So we often hear that enzymes are slow, they are expensive, you only generate low amounts of your product. Yeah, it's difficult to handle for chemists. I'm going to show a few examples to emphasize that maybe this is not true and we actually can use enzymes in chemical synthesis. So one challenge that we often see when we take enzymes from natural sources is that when we want to employ them in synthetic chemistry, the yields are often very low. That is true. So this is an example actually where we try to make some chiral multicyclic molecules and the yield is low. This is why it's important to actually engineer enzymes and improve them to make them fit for function. And we can actually do that in different ways. And there are techniques to improve enzymes in brief. One can use like a shotgun, you know, to shoot on the gene that is coding for the enzyme and then introduce random substitutions throughout this catalyst. Because as you know, enzymes are coded in the DNA, so we can actually mimic nature's own machinery to make mutations in this catalyst and to make new variants of it. So this is one approach. Then there is something called directed evolution, which is, well, it can also be actually error prone, but usually directed evolution is more focused on a few sites in the enzyme active center. And those are scrambled simultaneously. Then there is another approach called rational design, where you more or less try to understand what's going on and make single amino acid substitutions in the catalyst. So directed evolution was awarded with the Nobel Prize for its impact in chemical research. So it's a very powerful technique. And this is all now based on, of course, when we make new catalysts to be able to screen for improved function. I'm going to come back to that briefly. So if we compare a chemical catalyst versus an enzyme catalyst, like I say, they work with under the principles of chemistry, right? But what we can do with an enzyme catalyst is we can use the biological machinery to make new catalytic variants of this catalyst easily. It's possible nowadays in a research lab to generate 1 billion different catalysts and screen them by microfluidics. This is something that is done actually in, for example, in research laboratories at KTH or over, all over the world. So we can generate many different catalysts and actually analyze them in small format to find good enzymes, good catalysts. And this can perhaps be difficult to be doing by traditional methods. Okay, so I'm gonna talk firstly a little bit what we did with enzymes first in the Mistra SafeChem program, and then I will switch gears and say what we are doing now. Okay. So this is a little teaser actually for Ajay's presentation, but the first part of the Mistra SafeChem was motivated actually by the plastic problem. And as you know, this is a great challenge. And actually, one thing that you might not know, or you might know, is that plastics in textiles is a huge thing. So we made actually, if you're interested, you can read more in our review paper that was published last year. But we actually show that plastics in textiles will contribute to 3 billion tons of waste within the next few decades. 3 billion tons. That's tremendous, That's not, to me, not sustainable chemistry. And the first part of Mistra SafeChem, what we did at KTH, we tried to actually develop plastics that are bio-based and that can also be recyclable to actually try to counteract this problem. Now, one thing that is very important to notice is that even if we make bioplastics or biopolymers, that sounds great, yeah, but they can also face the same challenges as fossil-based plastics in terms of recyclability, etc. So that is important to say. But I will leave this also to Ajay later. She's going to talk about textiles. So I think actually this is also to give a little teaser for Ajay's presentation. I just want to say this market is growing, guys. We are talking about tremendous increase in production of synthetic polymers for textile industry. So this is something that I think we need to address rapidly. Okay, how to make biopolymers from biomass? One approach is actually to start what we already have now. So we look at what the fossil industry is making, okay, and then we try to make the same polymer, but starting from biomass instead, okay? That's one approach. It's something like mimicking fossil industry. But we are taking a little bit different approach. So I'm thinking like this, that we should learn from nature, because nature is not the same as a fossil industry. We should take advantage of the pathways that are already there in biology and use them to our advantage, to actually develop things that are in harmony with nature. I'm going to come back to that. That means that we're going to make chemicals and materials that have a different structure than what's out there on the market, but they have the same properties. And that will then make them, for example, recyclable. Yeah. So one of the things I've been focusing on in the first part of the Mistra SafeChem program is polyesters, because polyesters are highly important industrial materials, and they are also hydrolyzable. This is a very complicated slide. Essentially what it says is that if you look what's out there in terms of biodegradation, the only polymers that are biodegradable, meaning that they can degrade by microorganisms, is actually cellulose and pre-treated polyesters. This is pre-treated PET. This is like the half-life of these polymers by microbiological deconstruction. If you go to other types of polymers, such as polyamides, urethanes, etc., even especially the polyolefins, there is nothing out there. This is going to be a huge problem. So polyesters are good because they can in principle be degraded. So as starting material, so one thing of Mistra SafeChem is that we use biomass according to the 12 principles of green chemistry. And we start from biomass to make these materials. And we start, in this case, from a family of molecules called terpenes, which are really cool because you find them, they are natural molecules. You find them in, for example, in wood. So it's good also for the forest industry. Like I say, they have very interesting properties. They are made by really cool chemistry. So you start by simple linear building blocks that become elongated, and then they're becoming cyclized to form actually cyclic molecules with potent activities. This is, for example, limonene. It's the molecule that gives the flavor or how should I say, smell of citrus peel. So it's one example. This is actually industrialized also in material science now. Okay, so also it's very good to work with terpenes because they are cyclic, and when we make polymers with cyclic backbones, they give interesting properties in terms of stiffness and also, for example, glass transition temperature, because these rings are going to pack and interact with each other. So we can tune the properties of these materials in this way. And it's not easy to work, I mean, to actually do this, because we start with something here coming from biomass that's inert towards polymerization, because most of these molecules are not compatible actually with making materials. So we need first to activate them. And that's when actually we use enzymes, because enzymes can selectively upgrade molecules even from complex blends. Okay, so actually I talked a little bit about protein design. So I started, to do a structural-based protein design, but nowadays we are essentially only working with sequence-based protein and enzyme engineering, which I find very interesting. So we're talking about generating stable biocatalysts and stable enzymes. So one way I think we can do that is actually we can look on conditions on Earth, which are, let's say, extreme, because they're high temperature, harsh conditions mean stable enzymes. That must mean good business for industry, I'm thinking, because they will survive under, many turnovers. So I want to go to these conditions, and I'm very interested actually in looking on what's going on prehistoric Earth, especially, because then conditions were harsh, and we should find many interesting catalysts there, fossil catalysts that should be stable, because Earth had harsh reaction conditions under these times, and they should work well with industrial requirements. So of course we cannot travel back to these locations, but luckily we have the tools of statistical bioinformatics and sequence-based protein design. So we have actually developed algorithms in my lab. Also, other labs are working on this, I should say. But we can actually reconstruct the evolutionary history of enzymes and proteins and reconstruct fossil catalysts and bring them back to life in the laboratory. And they do amazing things. I'm just going to highlight a few examples. They are really stable, this catalyst, So we started with something, that had a melting temperature of 40 degrees, which is not so good. We actually brought the ancestors of this catalyst back to life in the lab. And we found that these guys, they were essentially difficult to kill, They had thermostability of 80 degrees. So, and we are talking about generating and screening only a few variants. So very, how should I say, worker friendly also, because we didn't have to screen that much. And we find very interesting catalysts actually. And then we are trying to figure out what's going on in the catalyst. In this particular case, we found that the loop in the enzyme was of particular interest for the evolution of this enzyme family. Talk more to me if you're interested about this, because I think I don't have that much time. So I will actually switch gears. And I'm just going to say that we work with chemistry. I'm also a chemist, so I'm not against chemistry in any way. I love chemistry, okay? But we are trying to combine chemistry with enzymes. And we actually did that in this particular case to generate then these novel biomaterials from terpenes, in this case alpha-pinene, a waste molecule from the forest industry. We could generate catalysts that can make key transformations, such as oxidations in water using oxygen as oxidant. This is a Bayer-Villiger reaction. In case there are guys from AstraZeneca here and chemists, I guess maybe you recognize this transformation. It's not so green, I would say, to do it in synthetic chemistry, but enzymes can do this with oxygen in water. And it's possible to engineer enzymes to make essentially only, high conversion under mild conditions. So we developed a toolbox out of this. This is what we did in the first part of Mistra SafeChem. We started from trees, we made new materials, and the materials are really cool, They look like this. They are compatible with different mechanisms such as polycondensation, ring opening, polymerization. They have tunable material properties in terms of TG, TM, really nice playground and starting point to continue with. And yeah, they are transparent as well, I should say. So they are highly promising bio-based polyesters for industrial applications. So yeah, we can scale this up, I should say. We can make this in gram scale. We can engineer enzyme plus reaction condition to make it 100% selective to only get one of the regular isomers. For those of you who are chemists, you know that's cool, I think. And another example, I will have my last 2 minutes then to talk about what we do now in Mistra SafeChem. I just want to show this. So we can actually, this is one of my PhD students, Jimena. She developed actually a microbe. This is the catalyst, guys. This is a cell. And what the cell does is it takes CO2, it starts from a hemicellulose-derived molecule, and it makes FDCA. And it looks like this. So I think this is something that everyone in the lab, even synthetic chemists, can work with. Because this is, I think we have pretty good yields. So we do this on the bench. We get a lot of FDCA out of this process. We are quite happy with this. It's nice for polyester manufacturing. This is what I did not talk about. And this is what I'm going to talk about for 2 minutes, if I'm allowed, because I think I started late. So we have not touched upon, or I did not touch upon the safe by design concept yet, which is what I'm going to do now. We have no idea if the molecules we make, if they are safe, if they are harmful. What about safety for workers in an industrial process? And this is what we are now addressing together with RISE, IVL, DTU, AstraZeneca, actually in an attempt to make fragment-based synthesis in a safe and sustainable by design fashion. So this pipeline is envisioned to be driven by stable enzymes that can make turnover after turnover under mild reaction conditions and producing molecules that are really cool and are safe. This is the key. They should not be harmful. So as a case study, we're working with amide bond synthesis because that's a very challenging reaction. It's not sustainable today. It's ranked as number 2 by ACS for chemical challenges in the world. So it's a big challenge, but we are collaborating and working with that. And like I said, I want to highlight safe by design. So I'm so happy we can work together with Ian et al. And work. You heard Ian's presentation. We're using all the fantastic tools he talked about. And we're actually, already before we make the synthesis of these fragments, we are making sure that they are safe. So Elizabeth is a PhD student working in the project. So we started with, in this case, with like 400,000 or 333,000 acids and 491,000 amines. This is quite big data sets. Now we are filtering them down by the Mistra SafeChem toolbox to make like safe and sound molecules that we know that they will not harm workers or persons that will then use them in products later. So in this way we can actually nail this down to a few molecules. And what we are doing at the present is we are down now in this cool project. At least I think it's cool. We are down to like 300 acids and 150 amines. And with this safe toolbox, we can build products for pharmaceuticals, for construction, for materials, hopefully, which are safe by design. So thanks I drag over time, I guess. Sorry for that, but thank you.
Thank you, Per-Olof. Yes, in the interest of time, maybe we should move on, but you will know where to find him during the intermission later or at the mingle, right? You will stay on. Thank you, Per-Olof. And we will move on to the third Mistra SafeChem speaker. Mathew, professor at Stockholm University, is participating on Zoom. Good to see you, Aji. You can hear us and see us?
Yes, I do. And do you hear me?
Yes, now we can hear you. Fantastic. So whenever you're ready, please share your slides.
Yes. Do you see my slides
Here
now?
we go. Please.
Yeah, thank you.
Yes.
So I'm really excited to be here. And first of all, sorry that I couldn't be there in person and deliver this talk. At the same time, I'm very much enjoying this session and listening to all your talks until now. So I would like to talk a little bit about sustainable textile recycling, some processes and products we are developing, and in the context of Mistra SafeChem. And as you can see, I have so many collaborators here, and which is one thing I want to emphasize from the beginning. This work is made possible because we have this group of excellent people coming together. Without that, this is not going to work. And so this is something we have all seen several times today. And here I want to just point out that I am focusing more on the materials and chemical building blocks, as well as recycling and reuse of the materials as such. So this is my primary focus area. And of course, it is supported by quite a lot of life cycle assessment and testing and screening of hazardous chemicals, which you will hear a little bit later on in the presentation. So coming to the question at hand, so why recycle textiles? So we have heard many arguments today why textile is something which we need to look at more closely. And as you can see, textile consumption is going to forecasted to be increasing, and it will be 140 to 150 million tons by 2030. And the most difficult thing about this is that this ends up in landfills or are incinerated or sometimes downcycled to other lower value products. And even 12% is lost even as without even a single use, as either as manufacturing of cuts or unsellable surplus stock. And if you look at the European scenario, we have at least 26 kilograms of textile per person is consumed per year. And from that, maybe almost half is discarded per person per year. So this is maybe, if you look really deeper, this is maybe abusive use of textiles as such. And another thing is, like, once in landfills, the natural fibers in the textile can take several years to decompose and has a potential to release methane and carbon dioxide into the atmosphere. So these are, I think we have enough reasons to look at this as a main important thing to work on. And also, as of today, only 1% of textiles are recycled back, and mostly into form of textile fibers, basically. So having said that, so we have an approach where we want to have an impact on this by converting this into nanocellulose. And here, of course, we want to move away from the current process where we are landfilling, incinerating, or exporting of this kind of post-consumer textiles. And the main motivation here is, like, textile contains a lot of cotton, at least 23% on an average. And this cotton is actually highly pure cellulose, which is a very valuable resource. And the hypothesis, therefore, is to extracting cellulose at the nanoscale from this kind of textile material can remove many of the defects in the original material, and we can get out very ideal building blocks for a wide range of high value applications. So basically what we can do with this approach is not to basically recycle, maybe even upcycle, because we end up with a product which is much more valuable than what we start with. So this was a little bit the motivation to go down this road. But in addition to that, we have, I have to go through this slide, because maybe when I say nanocellulose, many of you know already what it is, but still for those who don't know this field, so I just have put together a slide to show you what we mean by nanocellulose. So this is a product we get when we go down the hierarchy in a natural material like wood or plant cell wall. And here you can see that from the cross section, you will see the cells, plant cells, which contains different types of materials, which is hemicellulose, lignin, and cellulose. And we are having so many different methodologies already existing in our industries to take out hemicellulose and lignin, where we can take out just cellulose from the, cellulose. And actually cellulose is made up of a bundle of microfibers, which is in turn made up of elementary fibers as low as 3 nanometer in diameter. So this is the most elementary fiber you can get out from biomass. And this is made up of a chain of disordered and ordered regions of cellulose chains. And when we have this chain in 3 nanometer, we call it nanofibrils. Or when we take out the disordered regions, we end up with the ordered region, which we call cellulose nanocrystals. And you will hear a little bit about cellulose nanocrystals in the coming slides. So this is the type of material we would like to reach in the textile case also. And of course, the rationale for this is like we have a very parallel situation with the biomass, because we have textile also, which is abundant as biomass, and it is heterogeneous. Biomass is heterogeneous. And that is actually the case when we get post-consumer textile. We have wide variation in composition. And then in biomass, we have hemicellulose and lignin, which we need mostly to remove. But in textile waste, we have other polymers, which we need to remove to get back to the cellulose. And of course, if you look at biomass, we also have minor components and extractives like pectins, proteins, etc. And when we go into the textile side, we have dyes and other metal ions, which is in smaller quantities in the textile. So we have a very parallel situation, at least from my point of view. And if we fractionate this biomass or textile, we can get down to the nanoscale cellulose after a refining step as needed if you want to remove the other components. So this was how we try to approach this one. And moving on, so this is the first study we did, and where we have nanocellulose processing from textiles using the most traditional route to make cellulose nanocrystal, which is the sulfuric acid hydrolysis. And this, if you look at the first one, 100% cotton textile can directly convert into cellulose nanocrystals, because it is, as I already mentioned, it is high quality and high purity cellulose, and we get the cellulose nanocrystals. But if we take mixed fabric, we can do the same process with the sulfuric acid hydrolysis. What we end up with is again the cellulose nanocrystals and unaffected synthetic fibers, which is one route to recover the synthetic part also at the same time. And I will show you more examples in the next slide. So these are some of the examples or what materials we have worked with. The first one is a yellow fabric where we got, which is pure cotton, and from where we got almost 61% pure cellulose nanocrystals. And when you take the second one, which is blue CNCs, which is a mix of acrylic cotton, almost 50-50% in composition, and we end up with cellulose nanocrystals. And also on the side, you can also see the white fluffy material, which is the recovered acrylic material, which is still in the fiber form. We don't destroy the structures. And same thing with the red CNCs, when we start with polyester cotton, and we get cellulose nanocrystals and also the recovered polyester. And the last one is again a polyester system where we don't have a dye in that material, and there we get again the nanocrystals and the recovered polyester. So here I will say that we have two things happening. We convert the cellulose part of the textile into nanoparticles, which is having high value, and at the same time we recover the polymer part. And I don't have the data put up here, but we have looked at the molecular weight of the recovered polyester and acrylic, and there is no decrease in molecular weight, which means that we have not degraded those materials while we are making the nanocrystals, which we thought is positive. Then what else is there? So in addition to the nanocrystals, we could also see that there are some dyes there, as you can see from the color, and we tried to look at it a little bit closer using NMR. And the idea was to understand how much of the impurities is there. And you can see that the maximum we saw in the red colored CNCs with 2.2%. Others were like almost 0.3, 0.8, or 0.2. So relatively low amounts, but that also brings us back to the point made by Ian, that even in so small amounts, some of these chemicals could be hazardous, and we have probably zero tolerance on that. So this is something for us to think about when we consider the safety and the environmental benefits of textile recycling. And let me conclude this first one saying that this process was successful and we could get CNCs with high yield and high purity. And also this CNC showed the very typical behavior of a standard CNC colloidal suspension. But of course, we have this additional coloration in some cases. And we also saw that there are some residual impurities, mostly organic, though in trace amounts. And in some cases, it was actually close to the detection limits of the analysis. So also, if we have, you take a look at what is found in literature, we can see in the table, you see that summarized a little bit. And what we can see here is that there have been some works on similar systems from pure cellulose or pure cotton materials or dyed denim material, which showed similar kind of yields. And of course, there was not much work with blend systems. And also our studies show similar, yield and efficiency of the process. And in our case, the unbleached system in our study had actually much better process efficiency. And maybe we don't need to bleach and make it more, chemically intensive to extract this kind of nanoparticles and also extract out the polymer part of the fabric. So moving on, so this was the traditional route which we were following here. So we wanted to make the process more greener, or we at least thought we could make it more greener. And so we moved on to another acid than citric acid, which is a weak organic acid, highly soluble in water, and which is a solid at 25 degrees. And of course, we used it on 100% cotton and cotton elastane material. And this is the process we have done. Actually, this is a very similar process as we followed for the sulfuric acid hydrolysis also. So we cut the material and treat it with citric acid and at a high temperature and to make the hydrolysis, and then the reaction is quenched, and then we take out the non-cellulose part with the mesh, and then we use the filtrate material where we have all the cellulose through a sonication and a high pressure homogenization to convert it to nanoparticles. And what we end up with is nanoparticles with carboxyl groups on the surface because it comes from the esterification using citric acid. So this is again little bit images of that. So the first one is the starting material, and then we break down the structure quite a bit after the hydrolysis, and then after the homogenization or the fibrillation, we get nanocrystals, which is citrated in a suspension form in water. And if you compare it with the sulfuric acid nanocrystals, which you see in the first left image compared to citric acid, so we see very similar structures, and the yield was actually even higher in the case of citric acid CNCs, and crystallinity was actually similar in both cases. And also we had high surface charge in the case of citrated CNCs, which are all positive things to use it as a building block or a new material in different applications. And of course, in both cases, we have negative zeta surface charge on these particles, which you can see from the zeta potential data. So then I want to also go into a few things. Like this is earlier, all the work until now, what I showed you was based on attacking the cellulose part of the fabric and getting out the material. But then here we have another route which is developed by the Professor Joseph Samak and group, where we could also attack the PET part of the fabric in polycotton, and you can get, do a methanolysis to decompose the PET into dimethyl terephthalate and ethylene glycol in high yield, and which is a catalytic process which they have already developed, and then we can recover recycled cotton without any damage. which can be used either to make nanocellulose or even for a fiber spinning process, where we have tried to do it in collaboration with others to make it into new fibers. So this is also another possible route to, recover valuable materials from a post-consumer textile waste. Okay, so then other things on the other side, we also have to think about what is the implications of this kind of, recycling on the environment and also on the other hazard conditions. So this is why we looked at the residual impurities more closely by mass spectroscopy with the help of the analytical chemistry group, Ulrika Nilsson and her team. And what we see here is a volcano plot. I'm not an expert on analytical chemistry, but what I can explain to you here is that these are the chemicals you can see in the material. So left side of it is the starting material, and the right side of each plot you will get the product, the chemicals in the product. And also when you go more higher far from 0, 0, you get the concentration is higher and higher. So that is how you can interpret the curves or the graphs. And here you can see polyester cotton and acrylic cotton, and as well as the chemicals we have in the starting material as well as in the end product, which is the CNCs. And as you can see, there are a lot of chemicals there. And of course, there is less in the product than in the starting material. So, but at the same time, that doesn't mean that we have done a good job, because we have lost it in the effluent probably, which is also creating other possible hazards. So this is another thing which we want to look deeper into. And here the collaboration with Ulrika and as well as the collaboration with Ian will be useful and needed, because this is something we need to understand much deeper, the implications of these pollutants in different aspects of hazards. And then we also did some screening LCA together with the team at RISE. And this is a cradle to gate approach for laboratory scale processes which we have developed. And this is what we call a screening LCA. And we have 3 systems here: CNC prepared from wood pulp, CNC prepared from post-consumer textiles by sulfuric acid hydrolysis, as well as from the citric acid hydrolysis. So what you can see here clearly is that, if you compare the first process from sulfuric acid prepared from wood pulp to the cotton from textiles, you can clearly see that the blue ones have an advantage. So we are having a less environmental impact, and that is positive to use cellulose from textile as a source for making this kind of nanoparticles. But then when it comes to citric acid process, which we thought that it will be a more environmental friendly route, is actually not. It is causing quite a lot of putrefaction, especially in the terrestrial one. And this was actually eye-opening for us. So what we think as more environmental friendly and greener route is probably not the right way to go forward. And I have to also say that without this kind of LC and the possibility to look at it early on in the project, we would have never come to this conclusion. So, which means that citric acid needs to be looked at more closely. And that's what we are doing here. So there was a contribution analysis from where this impact is coming was studied. Or we looked at the different aspects like hydrolysis, quenching of the process, centrifugation, or other disintegration of the nanoparticles. And as you can see, the biggest contribution is coming from the hydrolysis part, and where citric acid by itself is the biggest contributor. So this was something which made us think again to see whether this is a possible route for scale up. And I think that this is a typical example how the environment like SafeChem will help us to make decision early on in the research to see how to go down the innovation funnel and make right decisions early on in the process. And therefore, we tried to improve the process by trying to see the possibilities to recycle citric acid. And this is some results from that one. And we found that it is possible to recycle citric acid by crystallization using little few drops of sulfuric acid and collect almost 58% of the citric acid from the effluent. And also it was obtained at high purity, and which can then be used to put it back into citric acid hydrolysis of the textile again. So this is one way to reduce the environmental impact or making the LCA more favorable for us. And I think we are running short of time now. So I would like to say that in conclusion, we are at a stage where we know that textile recycling until now has shown positive and promising results with respect to the possibility to separate textiles into components and recovering the cellulose fraction as well as the polymers. And we can also process the nanocellulose from the cellulose fraction, which is already a good thing. But evaluation of the environmental impact is crucial, especially early on in the research. And then one thing we are still starting to look at ongoing work is the evaluation of the effluent stream, which is equally important as the product. We need to understand what is in the effluent and also use that information to plan the scale up and experimental setup accordingly. So I think with that, I would like to close, and also as in the end, I would like to thank all the collaborators from Stockholm University, different departments, and also RISE. And I again want to say that without this big collaboration, we could not have done this work. And also I want to thank Varion Innovation, who was from the beginning and supplying us with all the post-consumer textile, which is sorted and which is provided to us. And with that, I would like to thank you all for listening. And if there's time for questions, I would be happy to answer it.
Thank you so much, Aji. We wish you were here, obviously, but since it wasn't possible, we're still grateful that you could share your ongoing work online. Thank you so much. Okay, it's now my great pleasure to introduce our next keynote speaker, Professor Lutz Ackermann from Georg-August-University in Göttingen, Germany. Lutz holds a PhD from Max Planck Institute and is now full professor at said university. He's also the founder and director of Yale Research Institute of Sustainable Chemistry. And Lutz's work focuses on organic synthesis and catalysis with a special focus on electrical catalysis and also bond activation. And now, Lutz, we're really looking forward to hear more about your ongoing work. Please.
Yeah, thank you very much for the kind introduction and also for the invitation, of course, Rika. And of course, also big thank you for all the other people that are involved in the organization of this exciting program. So, the first thing when you open the newspaper or listen to the radio is probably these days energy crisis, which is a different way of looking at a climate crisis to some extent. And therefore, there's a huge demand to move away from fossil energy sources, and certainly the use of nuclear power plant energy derived can be controversially discussed. Instead, using renewable forms of energy would be ideal and more sustainable. And within this context, potentially of a green hydrogen economy, it is not only about using, for instance, electricity from renewable forms of electricity for fuel production, but also to have an alternative and more sustainable way to produce chemicals in terms of power to X in a larger context. There are a number of challenges to be met to do so, one of which is in this larger context of moving from conventional forms of energy to green energy is obviously the supply and the demand of these energies, which is illustrated here for a typical day in northern part of Germany. Actually, I originate from Kiel, and I'm very glad to be back to southern part of Sweden. I was born in Kiel. So actually, I think there's a great perspective for these parts of the world to use renewable forms of energy. And for us, it's important in the context of how we can control energy conversion, how we can harvest, of course, renewable forms of energy and minimize, that is particularly important, energy dissipation. And we want to combine this with electrocatalysis that could be of importance in the larger context in terms of a decentralized green hydrogen economy. There are additional assets to this approach, one of which is cost of goods, which of course also here is of relevance. So if you compare green energy, and I will show you two examples, I hope, in which we use directly a solar panel or a windmill, the cost of goods for electricity compare favorable with different chemical oxidants, as well as typical chemical reductants. I hope it's readable, but these are the typical ones. I will today mainly focus on oxidants such as hypervalent iodine reagents or silver 1 salts. So electricity could be interesting in terms of using green and sustainable forms of energy, but also in terms of replacing chemical reagents, chemical redox reagents, thereby avoiding the byproduct formation that is associated with those. And I would like to emphasize this throughout the talk a little bit more. At the outset, we were wondering which might be the more interesting strategy. So I showed you on the previous slide, there are reductive transformation and oxidative transformations. And since we heard a little bit of CO2 use, power to X CO2 is of course very important. Let me share with you one example from our lab in which we do the electrochemical reductive coupling of CO2. This is under ambient conditions in which we can do these coupling reactions. And just to give you a flavor how we analyze these type of reactions, I brought you this slide. So we do typical electroanalytical chemistry, so CV studies combined with some inoperando studies, for example, here in this infrared spectroscopy. And then we arrive at a catalytic cycle, which I don't want to talk about in greater detail. Because we were wondering, do we study these electroreductive CO2 couplings? Or shall we do what we also like to do, and this is CH activation. And we chose to do CH activation for many good reasons. First of all, CH bonds are the integral part of any organic molecule, small, for example, drug molecules, but also, of course, macromolecules such as polyethylene. And by utilizing this strategy, we avoid all of these steps to make the pre-functionalized starting materials. And in one step, we can use this to do, for instance, late-stage functionalization, which I will talk a little bit about in the limited time we have here today, which could be of relevance also to the medicinal chemistry. And we're doing very fruitful collaboration, of course, with Magnus and his team here at AstraZeneca. But in principle, in all of these steps, we avoid chemical byproduct formation, and we can do in one shot the synthesis of the desired starting material. And we felt that this could be a nice strategical merger, because if you take a look at CH activation chemistry, so coming from the Nobel Prize winning cross-coupling chemistry, conventional method where you have two pre-functionalized starting materials, which is of course heavily used in the applied areas, you can either replace one, so in the electrophile, or in the nucleophile one pre-functionalization element. However, ideally, of course, you would like to do both, of course. To oxidative coupling, so a CH-CH coupling, and there are, of course, many important works which are summarized in these reviews here. And that would be the ideal scenario in which you basically have a cross-dehydrogenative transformation. So hydrogen is formed as the only byproduct. However, we have worked a lot on CH activation chemistry, and also in our studies, it was not the case. So what you actually observe is then indeed, most of the times you need a chemical oxidants, and very often that is a copper 2 or silver 1. And if you want to apply this, let's say on a ton scale process, that is a significant limitation. In that we had a Horizon 2020 project with a competitor from Belgium, in which there was an upscaling, and they chose a different route which used a silver oxidant, and finally in their upscaling they had a 25-kilogram reactor which was covered nicely with a silver environment and therefore difficult to use further on. So it's not only the cost of goods, it's also of course the practical procedure to upscaling. And I will come to an additional aspect later. So what we wanted to do strategically is to merge electro-organic synthesis with metal catalysis, and you can consider this as kind of an electron catalysis in terms of Amido Studers and DP currents definition, to merge these two to enable strong bond activation, which is not possible with simple electrochemistry, where you have, for example, the famous Schono oxidation as a typical example. There are significant challenges in doing so, and that is, for example, controlling, of course, energy transfer, and that particularly with respect to homogeneous catalysts. For example, if you have such a ruthenium catalyst, you see that you have preferentially the anodic oxidation. So we're talking about oxidation of an organometallic complex. It's an interfacial scenario at the electrode. However, you need to control over potential for the hydrogen evolution reaction versus the cathodic reaction of reduction of this metal. Within the Wöhler Institute, we're very much excited about this approach because maybe some of you know from the studies the Kolbe reaction. It's like the radical dimerization, decarboxylative dimerization. And Kolbe was actually a student with Friedrich Wöhler at the University of Göttingen. So there's a long history of sustainable chemistry in this context. Today, I want to focus on a few examples, conceptual examples that we feel are of relevance for this approach, one of which is related to this ruthenium chemistry. These are some reactions we reported previously with some citations. And I want to focus on this type of transformation where you cleave a CH bond and a heteroatom H bond with a ruthenium catalyst. It's an oxidative transformation. And so what you typically would like to do is take a look at the reoxidation event. Now, this is stoichiometric organometallic chemistry, and you can see that this ruthenium 0 half sandwich type complex can be reoxidized in an electroanalytical event. And at the same time, you see that the redox potential depends on the acid concentration for this elementary step of a catalytic cycle that then evolved in this system. So maybe a few explanations are worth mentioning within this optimization process. For instance, that it's fully tolerant of protic groups, so acidic groups such as this solvent system, which turned out to be best. We did not need a divided cell setup, so not a 3-electrode setup. That is important in terms of practicability, and we don't have a special additive as an electrolyte. So we have the sodium pivalate, which is used for the CH activation, which I can talk about more if you want to later. But as you can see, you get these alkyne annulations, and in terms of this synthesis, in one step, you can make these molecules with tolerating, for example, these heterocycles or reactive chlorides or esters, alkyl chlorides, and so on. Now that fundamentally allows to control hydrogen evolution reaction. I should mention so that the anode is a high surface carbon-based anode. The cathode is a platinum cathode, in which you go through this catalytic cycle. We can isolate and fully characterize these two intermediates as well as this one here, and the reoxidation I've shown you in an electroanalytical as well as experimental setup. But the key importance is that we now can couple this with a hydrogen evolution reaction, which could be utilized, for instance, for decentralized crop protecting agent synthesis or drug synthesis on demand. Maybe let me give you a few insights for what that could be useful beyond small molecules. We're interested in these larger molecules, polyaromatic hydrocarbons, and here actually was a combination of two very talented postdocs that came from Stockholm University, from Jan's group, who did great advance to make possible this electroorganic synthesis in which two reactions are combined. One is this Roda metallar electrocatalysis to build up these compounds. And if you're interested, the selectivity is very high. If you have an unsymmetrical alkyne, you can understand this by non-covalent interaction, so dispersion interactions. But you can also, and that's more important, combine it with a highly chemoselective subsequent synthesis. But let me show you the advances of this. So this is the Roda electrocatalysis with an aryl iodoboronic acid. And when you know palladium catalyzed cross-coupling reaction, you know that this with a palladium 0 wants to do a polymer. However, the rhodium catalyst, so this electrocatalytic reaction is highly selective. So you have high chemical yields comparably higher than with copper 2 or silver 1 oxidants. And you can combine it with the electrochemical Scholl reaction to make this PAH, which derive from linearity. Okay, but material science, maybe just one slide to finish this. Conceptually, we can make small materials that are of interest. Chemical selectivity. Selectivity is the point. I also think efficiency is the point. So it's highly efficient. You can have an iodide, fluoride, chlorobromo. You can have 6 hydroxy group in this annulation reaction. And for example, here you have a reaction that fully tolerates 6 azido groups. So you can do a click chemistry if you want afterwards. You can do Staudinger ligation, whatever you want with these afterwards. And another important aspect, I think, is scalability. So efficiency for scalability. If we want to combine this with the hydrogen evolution reaction, we need to have a scalable approach. And therefore, we probed here to combine this metallar electrocatalysis, so the combination of metal catalysis and electron catalysis with a flow setup. The specific reaction generates a number of compounds, either in an inter or an intramolecular fashion, which could be interesting for their biological activities. But in principle, this is now scalable, for which we used this commercially available setup. We have a 3D printer, which we modified it. So we created a turbulencer, a 3D printed turbulencer to improve the efficiency of the flow system, which of course then improves overall the mass and heat transfer. And it allows also to do some mechanistic studies just to show you that we're actually very much interested in how things actually work. So with this flow system and the isolation of all of these complexes, so these are CH-activated rhodium complexes. This is a 7-membered intermediate, highly reactive typically. You can now follow this by combination of metal catalyzed CH activation, electrocatalysis, and inoperando techniques, for example, the influence of the currents, so that is involved in the rate limiting step. You can identify these intermediates also by coupling with the NMR spectroscopy, and you can show that this is involving this key intermediate, which is oxidized in this case. Okay, just one example that we can not only cleave, activate CO2 and cleave CH bonds, but also CC bonds, which are of course the backbone of all organic materials. This is one reaction we had the pleasure of performing electrochemically, which is in the context of some work where we did some stoichiometric zirconium chemistry and some manganese catalysis. So you can cleave CC bonds, and you may ask actually why you want to convert this compound into this compound. Why not CH activation? Without going into the details here, it would be impossible to make this molecule by CH activation. And believe me, we have tried. You cannot make this Ricci isomer. So by CC cleavage reaction, you have actually a complementary Ricci selectivity here. Well, okay. So that is a short aspect of material science and CC activation. But let me come back to selectivity. Different aspects of selectivity that I would like to highlight here that go beyond the conventional mode of reactivity. For example, this metabromination, which can be done directly with HBr as the bromine source. And you may not get too excited with this phenylpyridine derivatives, maybe a little bit more for purine-based derived molecules. But what is remarkable, if you take these phenylpyrazoles and you apply almost any typical bromination agent, for example, this NBS, but other Br plus sources, you would get the reactivity at the pyrazole by an electrophilic aromatic substitution type reactivity. Here, without going into the details for the meta-selectivity, it's based on the ortho-ruthenation chemistry and directs para to the ruthenium in the meta position, but even on these very challenging phenylpyrazoles. So that's an aspect of site selectivity. Meta-selectivity, another aspect that I want to share here with you because it's a very fruitful, I usually don't talk about this because it's not finally published yet, but it's an example in which within this collaboration, we could address the electrochemical, so pallada electrocatalysis here without any directing group. So these are a little bit more complex molecules than you've seen before, highlighting some functional groups, and you get high site selectivity. There's a dependence on the electrode material, so if you're interested in this, you can find it on our homepage already. And we heard this beautiful talk before by Ein. So this is our first paper here, if you're interested in it, where we try to utilize descriptors and algorithms for also this specific question, but also beyond that in general for metalloelectrocatalysis. And this shows you that there is some selectivity. Actually, there's a selectivity that depends on the anode material without going into greater details and the time dependence based on two electro-oxidative transformations. Okay, selectivity. So this is without directing group, and this is now an example where we can show that you can do enantioselective electrocatalysis. So metalloelectrocatalysis in an enantioselective fashion. And what proved possible here is to use a transient directing group. And what we use here is this carbaldehyde motif, and this palladium catalyst can achieve high enantioselectivity, atropal selectivity, as you can see here, both for CC and NC axial chirality. And there are more and more molecules that become relevant also for applied areas that have atropal stable isomers. I would like to mention that here it's a palladium catalyst. It's a transient directing group, and please note that we have here a potentially oxidation-sensitive aldehyde, which is not oxidized to the acid, but which forms the imine and then allows this highly enantioselective transformations. Nice thing. This is another example that postdoc Johanna here did, where you can do allylation reactions by oxidative CH activation. This is an example where you can use a single palladium catalyst for two transformations. One is the electro-oxidative olefination reaction, and one is the reduction of the generated olefin that would have been here. And it's on the way, and it works to some extent as a paired electrolysis approach, where you would have 200% efficiency. So far, we are roughly around 100% efficiency in terms of using both half reactions. And what Joanna could also show is that you can do these reactions. So if you're interested in this, you can run these reactions on the balcony of the chemistry outdoor laboratory with a commercially available solar panel directly. So it's an example of an enantioselective metalloelectrocatalysis, dual catalysis, which is operative purely by green energy. You don't need to plug it in, and if you have an energy crisis or short supply for the PhD students in the upcoming winter, that's a solution you could consider. Late stage diversification, just I wanted almost to give a full talk on late stage diversification. It would be another talk where we do peptide modification and so on. So just one example here where we did electrochemical Late stage functionalization of tyrosine derivatives. Here you have a recent example where we also did a direct tyrosine modification with ruthenium catalysis. I want to share this example with you because you don't observe racemization of the backbone of this motif, and you can change the fluorescent properties of the tyrosine-derived small peptide for example, labeling studies. Okay, in the last final part, let me just shift gears a little bit. Another aspect of sustainability. So we try to merge metallo-electron catalysis with CH activation. But if you take a look at these metals, I mean, it depends on, of course, the amount of the catalyst you use, but if you compare the price, and that's the price actually relating to this review, which we did, the biggest review, wrote 261 pages. And okay, this is the cost of goods for typical metals such as cobalt, nickel, copper, manganese, and iron. And then you say, of course, that depends on which sector you're working in, if the price of goods for the metal catalyst is actually relevant. But potentially you could use, this is per ton of the metal, so you can use stoichiometrically these metals if you wish to. We heard already about the potentially endangered elements, but more importantly, particularly here for the pharmaceutical business, and I'm sorry that this is GSK data from a former PhD student of mine who's working in the UK for GSK. Obviously, I mean, as you know, the metals that I've shown you here, allowance of trace metal impurities is an issue, not only for the transition metal catalysts that I've talked about so far, so ruthenium or palladium or rhodium, but also actually the silver oxidant. So if you have a reactor that is plated with the silver, it's not only not very economical, but it's also an issue to use it further on for another process where it's getting contaminated with. So long story short, we're very much interested for a long time in using 3D transition metals, and we did so for cobalt, and that's actually in a reaction in which we showed that 3D transition metals, earth-abundant 3D transition metals can be used for metallo-electrocatalysis. There's a CO bond formation again to control the overpotential, so the energy conversion at the cathode. We have a platinum electrode and an RVC, so a high surface area carbon-based anode. And this is CO bond formation. Right, so it's a dehydrogenative CO bond formation, carbon heteroatom bond formation, which is tolerant. For example, cobalt doesn't like really to do cross-coupling reactions. It doesn't like to do Ullmann type reactions. So you tolerate an aerobromide or an iodide. You have oxidation-sensitive thioether, enolizable ketone, a nitrile or an ester. As you can see here, also in this work, we showed that you can use this not only to function as arenes, but also alkenes in a diester selective fashion. And that was one entry to ideally use renewable forms of energy. I don't go in this talk into the greater details of the mechanism, but this was our proposed mechanism, which we have a low valent cobalt, and I'll tell you more about this if you wish to, that is reoxidized. But I think strategically, the important part is that we couple it with a hydrogen evolution reaction. So now we can use electricity as a formal oxidant, replace stoichiometric amounts of silver oxidants and avoid the byproduct formation and combine this with a 3D transition metal in this case. And it's not only applicable to ether bond formation, you can also make esters, so acetoxylation reactions you can do. Amines I included here because in this case we had a program on biomass-derived solvents. You know, the major amount of what is in your reactor is probably the solvent. So this is GVL, lignin-derived solvent, and again you can do inoperando kinetic studies. CC bond formation is also possible. I don't want to dwell too much on the scope. Strategically, you have an oxidative alkyne annulation, and you have a electroreductive hydrazide here. The scope, again, aryl iodide, sensitive nitrile, but most importantly, I think from a strategic point of view is that we have here a triphasic reaction. So this is a reaction between the homogeneous catalyst at the surface, so the electrode surface, and combined with a gas. So you have a triphasic system, in this case, acetylene. And it's not only applicable to acetylene, this approach. We studied the mechanism, some DFT to check for some MS and some DFT to check for spin crossover. But what we could identify is, again, weak dispersive interactions to control the selectivity. And it's broadly applicable. It's broadly applicable. So you can use this hydrogen evolution reaction to form CO bonds, CN bonds, CC bonds with different alkynes, allenes, if you wish to, or CO as a gas. So at the beginning, I talked a little bit about usage of CO2 as a C1 source, and here you can use CO for this electrochemical transformation. Well, you may say, okay, I had one example for reactions without directing groups. So this palladium electrocatalysis. This is complementary in a way that it works without directing group and it works on simple alkanes. So before I talked arenes and alkenes, but as you can see here, this manganese electrocatalysis works preferentially on the benzylic position over the aromatic system, but you don't need the benzylic activation. So you can use cyclooctane. And this we're heavily investigating now for instance, for polymer upscaling, but also other purposes such as late-stage functionalization of some drug-relevant molecules. Now, this you can use to modify these, compared to the previous slides, slimper molecules in a late stage. One step, you introduce an azide, you can do click chemistry. And if you're interested in the mechanism, it's based on a manganese 3,4 catalytic system. That brings me sort of to the end, but I included a few slides because all the time I was debating, do I talk about electrochemistry or do I talk about peptide modification? So just to give you 3 slides, sort of what else we are doing. So we're interested in these 3D transition metals, and here we made some cyclic peptides, which we studied for the cytotoxicity with our university hospital. Here we developed, so if you're interested in labeling, we have reported on some BODP modifications and NBD labeling, and together with Mark Van Drell, we used them for in a new amino acid that could be used as a cholesterol sensor in the cell. And last but not least, of course, this is the most important slide, and I'm glad that Stick is here. Beautiful work, but I don't need to discuss it in greater detail because you should know what it's about. It's a very exciting and fruitful collaboration, which we can do direct the modification of all of these complex structures. In this case, magic methyl effect, and hopefully soon also the isotopically derived molecules will be available to the public if you're interested in this. In the discussion, I could talk about more in the mixer. So with this, I just want to mention that we're also interested in some research with other researchers in the world, such as Chagas disease or anti-malaria, for non-profit organizations where we make some new hybrid molecules for by CH activation. Okay, coming back to the take-home message. So we combine metal catalysis with electrosynthesis. And this is the one example, or the earlier examples in which we actually did this here. And this is the setup, as you can see, so it's outside. And this is the reaction which we specifically did on the shortest day of the year in 2018. So this was before the pandemic, in which we used this commercially available solar cell, but also this windmill here. So you can buy them, it's relatively cheap. You can use it as the sole external energy source for the PhD students, again, if you have some problems running the reactions in the winter. And thereby be self-sustained. And maybe one additional aspect that I want to make, we are also interested in photochemistry. And in addition to the inherent limitations, maybe one aspect you want to consider if you use electrocatalysis or metalloelectrocatalysis, if you have a specific redox potential where you want to do your reaction, For the photochemistry, if you want to move from 1.2 to 2.2 volt, you need to synthesize a new photoredox catalyst, and you need to buy it, and you need to make it. In contrast to that, with the potentiostat, you can dial in the potential of the reaction and therefore select your chemoselectivity. With this being said, I hope I could give you a little bit of glimpse of the electrocatalysis we're doing, where we try to use green, or we are using green energy, 3D transition metals, lignin-derived solvents, and we try to control all elements of selectivity, chemoselectivity, site selectivity, and enhancer selectivity. And we hope that we can contribute with this, not only replacing chemical oxidants, but ideally to having a decentralized green hydrogen economy of the future. Okay. Well, this is a different version of that. Yeah. This is a catalytic cycle. I would like to thank you for your kind attention. That's a different version. Yeah, beautiful chemistry. But this is actually the end of the talk. So thank you for your kind attention. I would like to thank, of course, my coworkers for their dedicated contributions. This is before the summary slide. So, well, okay, there we are. I'm grateful for all of their experimental work, but not obviously mostly for the intellectual contributions. I'm grateful for these fruitful collaborations and these funding agencies, and particularly also here with Magnus and the team. And last but not least, you for your kind invitation and attention. Thank you.
Thank you. Thank you so much. Are there any questions in the room? There's nothing on the chat. We checked. You will also be here for this, so there's plenty of opportunities to ask questions also during the intermission and the chat later on. There is actually time now for a short intermission, but with short, I really mean short. So don't go anywhere. Please feel free to stand up, stretch your legs, say hi to your neighbor or something, but don't go far. We are saving a really nice treat for you in terms of a lecture after the break, so you don't want to miss out on that. So please be back now by 4:20. Okay, 4:20.
It's my pleasure to welcome the last invited speaker for today. So we have Stewart Owen, who is the principal environmental scientist at AstraZeneca. He also holds an honorary associate professorship at University of Exeter and has a training as an environmental toxicologist, if I understand it correctly. Yeah. Please, look forward to your talk.
Thank you very much. Thank you for coming back, everybody. Firstly, I'd like to share an apology from my boss, Jason Snape. He sends his sincere apologies that he couldn't make it to come to present this. And I'd like to thank him for giving me probably the longest title of the day for any of the presentations. I'm going to try and break it down into 3 pieces. And I'd also like to try and go a little bit quicker than I originally planned, because I think the mingle is really important for us. But we'll talk about the discovery and development, then our patients, and then talk about life cycle as well. And I'm going to get there. I should have said at the very beginning there, please feel free to take out your phones, and rather than trying to type down links and things, I thought I would put some QR codes for you to take to places. One of the challenges I get a lot on that gap between academia and industry is, how do people find out what industry are doing, and what have you done? You know, how can you do these things? Most companies, most of these places produce lots of big reports, lots of people behind them to try and do that. This will take you to the 2021 sustainability report from AstraZeneca. There's lots of pretty pictures in there. There'll be a new one next year. There's probably 10 years of them sitting there on the website that you can go through. And while you're getting your QR code out, this is me sort of not being sustainable, I suppose. So if you go back 120 years, when bicycles didn't have brakes or chains, they were still bicycles. And what I was trying to do with that, apart from point out that I believe in progress. The next generation are obviously much quicker, and this chap here is about to overtake me in this race that I didn't know was going to be a race, a bit like this talk that I didn't know was going to be a talk. So hello to everybody on the Zoom as well. How do we get to this place? And for me, I brought my Bible with me, and I didn't steal it from a hotel. This is a 60-year-old book. That's the first edition of this one here from Rachel Carson. And I don't think it's the very start, but it's probably close to that. And I was going to do some quotes, but we won't do that because it'll be a bit quicker. 60 years, sorry, 60 years is an awful long time to be still talking about the same things. And you got to remember it took 22, 23 years before the legislation changed, basically on the backs of that book. We've now got things that are probably bigger and more front of people's mind at the moment, things like climate change. Well, 60 years ago, some people knew about climate change and some people knew why it was coming through, but it wasn't a big topic. So there's another little QR code there that will take you to the IPCC report. That's a massive report, as I'm sure most of you know. And that's talking about the climate side of things. And then there's another one over here from the Lancet Commission on Health. And that's another little separate link that you can take to that one. And that one will tell you about pollution and the effects of pollution. And so pollution's huge. Lots and lots of people are dying from pollution. The impacts on society, the impacts on different parts of the world. It's a really big piece. And that's how I got into ecotoxicology and back into that side of things. And this is my final slide, he says, in a couple of minutes late. There are more. I thought I'd show the last one first, because what I'm trying to say really is, I think that all of you have one piece or several pieces or big pieces and small pieces of a jigsaw. So whether you're coming at this from being a chemist or whether you're coming at this from a biologist or just a concerned person, I think we need to be able to start putting these things together, and a lot faster than the 20 odd years it took to get to that, and the 60 odd years we're still talking about it. So that's my final slide. Let's go back to these three pieces that I thought we'd break it back down into. I can very quickly go on to these. So in that sustainability report from the very first QR code that I gave you, you'll find pieces like this. And the whole point about that is it takes big teams to do this thing. And we're not brilliant at it. We don't all know what we're doing about it. Some people clearly do. Nobody explained it all to me. But actually, we're doing our best to try and do that. And so are lots of other scientists that are industry-based, as well as the academia and as well as the regulators. They often get left out of this, that they're all working in this area. So in terms of discovering and developing a new medicine, everybody's talking about greener, greener design, a greener process, a greener manufacture and distribution. Each one of these areas are really difficult to deal with. Really, I'll come to those in a second. We'll get through these pieces. But what some of the things we've heard today about using different metals for your catalysis, well, that's all part of those pieces. I think I'd probably rather you were using iron, not because it's cheap, but because there's lots of it. And the actual mining of it per kilo, ton, whatever you want of that, is probably a little bit easier to control than going after a ton of cobalt or a ton of palladium and into those sorts of pieces. So it's about that holistic piece and trying to understand what you do now. Is that the best way already? Or do you put effort into changing the way you do that? So if you've got things like, let's say paper. So the supply chain for paper, we've got a fairly good handle on. We know where the papers are coming from. You know how to get a greener supply of that and to include it into the recycling things. We might not be really good at it, but we know quite a bit about that. Is that where we should be putting lots of effort? Is that the biggest hit? And that's the part that we'll link through to in a second in the life cycle of trying to understand those pieces. I've been involved in a very large consortium project. We were lucky enough to get a publication in ES&T Letters, Environmental Science Center. So it's an American Chemical Society journal, and it's got the editor's open access award to it. So you'll be able to find that on a free and open. That's the other part we're saying about trying to get that communication out about what's available. I'll leave that QR code there. Well, it's the same QR code, but I'll just try and make those words a little bit bigger in there. So I'm not going to go, in the interest of time, through each of these letters through greener. You know, it's a nice acronym that we went for, because G is about patience. So we're trying to struggle to find how do you put patience into greener. But actually, pharmaceuticals are really important and are slightly different to other chemicals in that you're always looking at the benefit and the cost benefit analysis and the risk benefit analysis. But at the end of the day, it's about population and it's about individuals and the difference those make. So that's your starting line on there. And then if we jump down things on here, let's take the second E there. It's about environmental biodegradability. We talked about that, bioaccumulation and those things. How do I explain to a medicinal chemist, not being a chemist myself, what properties are going to be really useful for a pharmaceutical after it's been through the patient? And when it hits the environment, if it's going to hit the environment, what do we need to design in? What do we need to design out? And that's what this little, it's a very short paper, there's only a couple of thousand words in there. And we talked about other bits and pieces in that paper that I think you might find quite interesting. It's a very easy read, at least I thought it was an easy read. And it's grown out of a project called Premier. Now, Premier, I'm the industry lead for that. There's 25 different partners. There's lots of people. When I say I'm the industry lead, I'm the guy at the back holding onto the reins and everybody else is doing all the work in the front. And there's lots of different projects that have touched on the talks that we've heard today. Other people in other arenas trying to do the same things. So what tools does a medicinal chemist need in order to understand the environmental risks? Should we be using some of the Daphnia work that you've heard from the posters? Should we be doing the in vitro, the machine learning, the AI type production? Well, they're all within Premier. So Premier's got 64 years of effort wrapped up in it. There's 10 million euros. I should know more accurately than that. Maybe lose a million here or there. And there are more partners joining that as we grow. So there's another 2 partners coming down the pipe to come and join and put more innovation in that. So there's another 4 years left on this. We're 2 years in. It's a really exciting project. That QR code will take you to the website to have a little look more of that and to see whether there's any tools there that you can start to use as well. Back to my 3 circles. I was talking a bit about patients as well, wasn't I? It was about them being important on the green. And I think one of the things we tend to forget, particularly in a Western world, is about how many people don't have access to medicines. And you know that number is really hard. It's 2 billion, really hard number. And that access to medicine is really one of the key pillars of how do you help the global population with this. And you know, so we talk a lot about sustainability, and that second line there says least sustainable pharmaceutical is the one that doesn't get to a patient. And there's a whole project about the last 10 yards, last 10 meters. You'll find lots and lots of that on the internet for those sorts of things. The other thing I think is really at the top of my mind is pharmaceuticals are here for a long time. So probably most of the pharmaceuticals that we have on the market at the moment are old. An awful lot of them are older than this 60-year-old book. So your grandchildren, and probably your great-grandchildren, will still be wanting those medicines. So how do we manage how you manufacture them? How do you get them to the whole global population so they all benefit from it? And how do you phase in greener ones where it's important to do that? And how do you choose which ones are most important? And how do you know which one of the ones you should really worry about when they are in the environment and which ones you should worry about less and that there are other things that you should prioritize? That's a lot of what that Premier project's about. We've heard today quite a lot about how you do some really cool chemistry. Most of it is somewhere about there on my understanding, but I need everybody to do that in order to help how you get this for the whole of the medicine industry, the whole of the pharmaceutical industry. How do you get really good at doing these things? Which solvents do you want to use? Which solvents don't you want to use? Are ionic liquids the thing to work with? Or are they not? It's really important to understand that. And I've heard this quote across the bottom here, and I've read this quote, but Google disagrees with me. So if Mistra wants to try and grab hold of that, I think once we've dealt with climate change, I think we're still going to have pollution problems. I don't think this 60-year-old problem is gone away. People are still going to be taking medicines. Some of those medicines go through, some of those medicines end up in the environment, and that's a really important part of that. So what can industry do about that? Well, I know that there's analytical chemists and environmental analytical chemists that go out with their sampling all over the world, and they measure the pharmaceuticals in the environment, and then they report them in the literature. So what we've been doing is we've been collecting those. So 32,000 measurements, sometimes not all of them are detects. Some of them are below detection limits. Some of them are not there, some of them are there. We take it at face value and we've put it onto this app. So if you don't click on anything, this is the one I'd really like you to click on and have a little look at what the distribution is. This is just AstraZeneca's medicines, it's nobody else's. There's a risk quotient allocation to there. It's an interactive thing. You can zoom in, zoom out. So obviously, you know, I'm pandering to my audience here. This is a global thing. So you can find them in the Antarctic, you can find them in the Arctic, you can find them just about anywhere that anybody wants to look. Wherever people have been, there'll be pharmaceuticals there to be able to measure. And analysts are really, really good at finding them. So which ones should we be worried about? And so this tool lets us start to do what risk assessors do and work out which ones we should be most worried about. And in our case, there's probably about 70 occasions where either in effluent or in different matrices, you're finding risks that are higher than one. So that you would expect there to be if what says in the lab translates. Hopefully you've got those on there. Let's jump ahead now to life cycle. I'm conscious on this time. There's an awful lot talked about life cycle and how you would get into that. One of the things that I think is critical for people to understand is how difficult it is to make medicines. Really, really hard to discover them, really, really hard to make them work, and very, very hard to get them to made and out to the pharmacies and to the patients. How we do that and the effort we put in that is enormous. Large pharmaceutical companies probably have a first tier of suppliers of, say, 50,000 different companies that supply them. Big pharmaceutical companies might have 150,000 suppliers, but everybody's different in how they do that. How do you know what their suppliers were doing? Where did they get that material from? How do you follow that chain down? When people ask for a transparent approach to where these medicines are and where they've been manufactured and things, it's complicated. No one place makes one medicine. No one medicine is made in one place. You have different chains running because what's most important is the other slide with patients and trying to keep that guarantee of supply. And when you put all those together, this is the sort of slide where it says something and nothing, I suppose. What I'm trying to find is that sweet spot, that piece in the middle where you can manage and understand your footprints, you can understand your manufacturing and your distribution, and you start to look at what resources you've been using and understand the circularity of bringing those resources back around and pulling those together. I'm very near the end of this, and I just want to, so in that sustainability report, we point to all of these different pieces. That EPV, ecopharmacovigilance, so medicines are monitored by clinicians and side effects or off-target effects are all reported back in through a process called pharmacovigilance. And what we're trying to do as part of that is for the environment, we call it ecopharmacovigilance, and our team are trying to manage that. And that app that I was showing you with the GIS mapping and things like that, it's all part of that process. Told you it was my last slide. So it's the same slide as the other slide, except this time it's got a link there for you that will get back to me. And I'm very happy to take emails or contacts or however you want to do with those. I'm also keen that hopefully we've grabbed a few minutes and there'll be a question or two. Thank you.
Thank you very much, Stewart. And yes, we have time for questions. Yes, please.
Yeah, Stewart. There you go. Press on the. Do you hear me?
Yeah.
In some countries, we have laws regarding manufacturer responsibility and recuperation material. So in this country, for instance, we like to collect aluminium cans, or we like to have some industries that will collect their products free of charge. What's the status today of the responsibility from societies with respect to the pharmaceutical industry about its waste? You said discharges of API, but also in hospitals, patients. So what's the status?
The simple answer is very messy. So it depends on which region you're at. So in some regions and some localities, you have take-back schemes where unused medicines go and you can return those. Largely, they go for incineration because you've lost the chain of command for those about how they were stored and how they were looked after and all the other pieces in there. So that's one mechanism for pulling that in. The manufacturing, the industry is working very hard towards controlling its outputs and its controls on there. So we have a manufacturing discharge. One of the slides I skipped over there was around our own sites globally. We monitor and predict and measure those things, and our suppliers report into those. And because of that complexity of the supply chain, it's very hard for bigger companies than AstraZeneca to do that, but they're trying, and that's coming round on there. And then a lot of this is also about where do you get your medicine supply from. So most pharmaceutical industry is really odd. So you have innovation companies that have a very small number of products still on patent, and then you have a very large industry of generics where you have the more common things, the paracetamol and the ibuprofens and those sorts of things. They're harder to understand because it depends on the purchasing process of the regions that are actually buying those medicines in the first place. So if they're only interested in price, they go after one thing. If they're interested in setting standards and want the manufacturer to meet certain standards, that goes them down another road. This whole world is moving along very quickly at the moment, and lots of regional medicines agencies are starting to pick that up. And I'm really hopeful that that's going in the right direction that we want it to. But it's messy.
Yes, we have a question.
Thanks, Stuart. Peter Vanke, DTU in Denmark. Excellent talk, and it reflects on the challenges that all the companies have in the world on the chemicals and pharmaceutical sectors, starting with complexity in supply chain management and so on. When we step back a bit, I would like to challenge when we want to really facilitate a transition out of that mess, as you call that. When I think 100 years or more, the whole market worldwide has been optimized for one thing, that is economic sustainability, efficiency. So I make the link back to Anastas in the morning. What we are not able to do within the frame of the market as we have created it ourselves is effectiveness towards the social side. We don't reach the patients where they are. And the environmental side, we cannot produce within the environmental boundaries currently. That's what you're essentially seeing as a challenge, right? So how can we get out of that? Because this transition really requires, I mean, nature doesn't optimize for money. And I understand that it's essentially coming down to the question, how do we finance that transition? So what are your thoughts on this? Thank you.
Yeah. When Paul challenged us in his talk about efficiency, I thought it was a really nice way to actually get at the economics of this situation and separate out that from the needs of what we need to be doing. I think most of us are sensible enough to know that money makes this world go round. How do you therefore apply the value to the environment so that the environment actually gets its benefit as well as your patient benefit? And I'm skirting around the idea is, I don't know. And anybody that can tell me how to do it, please do, because we'd like to. We're pushing towards these changes. You can see some things are coming in faster than others, but this period of transition at the moment is really difficult. And if you throw in the mix the instabilities in the world created from the effects of the COVID, from effects of war, from effects of all the other pieces, you know, where does this sit in people's priorities? Are there other chemicals and other things that people should be more worried about? I don't know the answers, but it's difficult. We're trying.
I think that will be the concluding words from Stewart. Thank you very much again. We come to the final words given by Richard Lihammar, the program director of Mistra SafeChem. Please.
Sure. So, can you hear me? Great. People joining from home or work, I need to leave at 5, and people here probably ready for mingle. So I say like Henry VIII told his second wife, I will not keep you for long. But to start off, I would like to thank again the speakers for today. Malin, Paul, Ian, Per-Olof, Stewart and Lutz. I think they deserve one more round of applause. And also thank you AstraZeneca for hosting us today, Magnus, Christina, and Malin, that you helped out with the planning and fixing today, and also the organizing committee at IVL, Hanna, Ragnhild, and Lennart and Ulrika for fixing this wonderful afternoon on green chemistry. And then I figured, since we have some minutes to spare, and this is a Mistra SafeChem event, it would be nice to talk about me, the program director of Mistra SafeChem. I am a chemist, and that means that I'm a superhero with a superpower. I can make almost any molecule in the world. If you give me a good lab, some students, analytical equipment, and some time, I can make them. Like Lutz showed, you can make any kind of molecule really in quite good and efficient ways. And this goes back to what Paul said. I mean, chemistry is essential and important. Few of the advancements made could have been made without chemistry, like renewable energy, medicines, as we hear at AstraZeneca today, agriculture, technology, energy storage, and also various materials such as plastic. I mean, chemistry is needed, and this innovation process needs to continue for us to keep on evolving and making also the sustainable side for the future. And one part of Mistra SafeChem is to keep on doing these inventions. But as we know and talked about today, there is a backside as well. First of all, it runs on fossil-dependent resources. And that's one thing, this climate neutrality needs to come, but also the pollution. I mean, things that should not have been made and spread in this way has been made and spread on a global scale that should have happened. Like lead in gasoline, widespread use of insecticides such as DDT, as these American soldiers are testing out. And also the latest scandal, perhaps with PFAS, for example, shows that chemicals should not be used and spread in the way it has been. Now, using the kind of negative examples, maybe not the best case to show what we should do, and maybe puts the focus on wrong things. And my idea of looking at this is, would we have found these things today if we would be in an R&D chemist lab? Of course, these things we might find because we know about this now, but the new problem, will we know about them? And that's one part of Mistra SafeChem, to be able to find these issues already in the design phase and see if we can do something about them before they are launched and they are spread. So we are a group of chemists, toxicologists, ecotoxicologists, life cycle analysis experts that try to collaborate to enable a safe and sustainable chemical industry. And this collaboration is key here. I mean, these disciplines usually do research, tremendously good research on their own. But here we try to put all of these different functionalities in the same room around the same table and look at the problem together so that we can do it early in the development phase. So one part of the course that's new and sustainable synthesis processes like Per-Olof and Adji presented, new ways of making molecules in a good and innovative way. But we also like to add on a toolbox that can predict and avoid risk already in the design phase. You touched upon it, Per-Olof, that you can use in silico tools to screen your start materials, take away those start materials that are not interesting from a hazard point of view, focus on the ones that are regarded safe, and from there start building your process. Adji could find that she had two ways of doing her synthesis, either with sulfuric acid or with citric acid. By the use of the toolbox, she could see that potentially the sulfuric acid would have been the more preferred choice for this reaction. Now, probably without this toolbox, she would say that citric acid is better than sulfuric acid, publish a paper saying we have a green process for textile used in new kind of materials. But with this kind of toolbox, when evaluating the process early on, she can get flags. Is this the best way to go about? Or perhaps both routes are bad. We should find a new way completely. And this is what we're kind of trying to do, to get out research that are safe and sustainable by design. And this safe and sustainable by design, maybe you heard about it, some of you, that's really a key term now in the EU, ongoing research in EU. For you who don't know it, 2 years ago approximately, the EU launched a strategy called the Chemical Strategy for Sustainability. And this sets out a roadmap, a route, how to really reform the chemical handling in Europe. One part is increased legislation and how chemicals can be spread. That's kind of the restricting side of the chemical strategy. More interesting for us is the allowing side or innovative side, where they highlight that, yeah, we need innovation to drive this. We need good innovation, also because we need to tackle all the issues that will come in the future. So where things have spun out from this is like the PARC program, Partnership for Assessment of Risk from Chemicals. And I put it here because we have links to this program. Tools that is needed for this assessment could and will be developed in Mistra SafeChem and could be transformed into this program. In the middle, we have the Safe and Sustainability by Design framework, which would be like a new way to innovate chemicals and materials, and to have like a framework that you can go through step by step to see if your compound material is safe and sustainable. But this is hard. I mean, we're not there yet. And part of Mistra SafeChem is to give those tools and processes to make it safe and sustainable by design. And one way that shows that this is important is that EU is pouring funds into this. I mean, there are several Horizon Europe programs coming out now that goes in line with the Mistra SafeChem program's ambitions. And we're happy about that because, as I said, this was launched 2 years ago, the strategy. Mistra SafeChem started 3 years ago. So we actually 1 year before this strategy was released. So it was really forward-looking for Mistra to announce this call. And it also means that we have started this process. We have our case studies that are running, looking at these issues, and we hope to play into these ongoing activities that will happen during the coming years. And I said Mistra, they are the funder of this. As Ulrika showed this morning, majority of funds come from Mistra, but also the participating partners come with funds. We're halfway through, and for our first phase, and the second phase might go until 2028. So this is really a long-term investment for Mistra to make this network and do this research in Sweden on sustainable chemistry, which we are truly happy about. So any playing of needs or problems that you see on your side could be played into us, because we would love to see which problems are out there and try to tackle them in our current and future research. And who are we? We're 6 research partners and 13 companies. Me, myself, I'm not from Mistra, but I'm from IVL, Research Institute, focusing on applied science for a sustainable society. I'm work at the Stockholm office, but we also have offices in Gothenburg, Skellefteå, Fiskebäckskil in Malmö, as well as India and China. And we are the host for Mistra SafeChem. But we're not doing it alone. As you've seen today, we have a representative from Stockholm University, RISE, KTH, who present today. Also Peter from DTU is part of the program, and also Chemsec doing research. But not alone, we do it in collaboration with the companies as well that are joining the program. Here depicted on the logos, now in alphabetical order, but if you wash them around, you can see that if you put them in another order, we kind of get the value chain for chemicals here, starting with producers like BASF and Perstorp, also Holmen and Renfuel that make compounds from forest residues, over to the product manufacturers, such as Astra being here today, H&M, Cytiva, Volvo, and then the end-of-life process or recycling processes also we have represented. For example, Vargön that provides Adji with textiles for her project. And here we learn from each other, trying to see the best practice from industry, like Stewart said. I mean, your process that you have in-house could also be used for other industries and other participants. So where we can collaborate and share knowledge, we try to do that in order to move forward faster. And so far, we have had some numbers which we have achieved so far in 2 years. The number one and the one central one is this new network. We actually work together, these different disciplines now, trying to make new science, learning and teaching each other on each other's different subjects. Around 92 people working, published 44 papers in these 2 years, held some external seminars, which you can watch on our webpage. We have 11 case studies running where we try different synthesis route and try to apply our toolbox on it. Adjis and Per-Olof are 2 of those. Held 2 program conferences. This is the second. And this will continue tomorrow for the internal group where we'll keep on developing what we should do in the coming time. So some of you will stay around for tomorrow as well. And we also have a research student group where the PhDs and the postdocs of the program meet and present for each other. And also have some social activities, if I haven't heard wrongly. So we try to really put these different PhDs from different backgrounds together to learn each other. And with that said, it's drawing to an end of this seminar. People who joined us digitally, we're happy that you've been here. Thanks for all the questions. We have not been able to take any of them so far, but we'll try to answer them in retrospective on Zoom. And if you do have any comments or need to contact us or any of the speakers, you can find details on our webpage. And for you here, now it's time for a longer break, so you actually can speak to each other a bit longer. There will be snacks outside. There will be some drinks. And if you need something stronger, also the bar is open if you want to purchase something from there. And last issue for those joining for the dinner tonight, sorry, this is only Mistra SafeChem participant. The rest of you, close your ears. The bus leaves at 6:30 from where you entered. And that will take us to our castle where we'll have the dinner. Not maybe this one, but a similar one. And keep your name tags for tomorrow. Otherwise, AstraZeneca will not let you in. So you have to keep that and bring that tomorrow. But with that, that's all for our program today. And I thank you all for coming and joining us. Thanks.